Hello, can you hear me?
Yes, we can hear you.
පටන් ගන්නද? Okay, a very good afternoon to you all who are joining here with us in this online session number 2 of the International Open University Research Sessions 2022. Please keep note that you are now in parallel online session number 3 for biological sciences. Today, we have 9 presentations with exciting topics in line with this session, and we are about to start the sessions from the first presentation up to the 9th one. And let me first introduce our chairperson for session number 3, Biological Sciences, Professor Tamara Veerasinghe. I cordially invite Professor Tamara Veerasinghe, the chair of the session, to take over this session and carry out the rest of the procedure from now onwards. It's over to you, madam.
Let me, as the chairperson, again welcome all of you for the International Open University Research Sessions 2022. I'm very happy and I'm honored to be the chairperson in this very valuable session. Right? So let me tell you, there are 9 presentations lined up for today's session. As you heard earlier from the Dr. Ruanile Perumal, this is the theme is biological sciences, right? And each presenter will be having the 15 minutes time total duration. Out of 15 minutes, you will be spending 10 minutes for your presentation and 5 minutes for the questions and answer time. So I cordially invite all of you to participate in the Q&A session, which will be very valuable for the participants. So let me first introduce the first presenter, ID number 25, determination of the antibacterial properties of endophytic fungal crude extract isolated from Acrosticum aureum. The authors are Kavinya Rajan, F. Asma Rahman, and D.M. S.U. Disanaik. The presenter will be Kavinya Rajan. Over to you, Ms. Kavinya.
Good afternoon, everyone. I'm Kavinya Rajan, and this presentation is about the research on determining the antibacterial properties of endophytic fungal crude extracts isolated from Acrosticum aureum. Antibiotics are products of microbial synthesis that inhibits the growth or kills other microbes. Antibiotics have gained an enormous development over the years from the usage of mildewed bread or soil to the current synthetic agents. Yet, antimicrobial resistance has risen as a global crisis which is not given much airtime by public in general. As Figure 1 represents, a Lancet review and a UK report predicts its impact as greater than many infectious diseases in the future. Hence, scientists are now designing and new antimicrobial agents using the secondary metabolites, either synthesized by the plants themselves or by the endophytes residing within plants. Considering plant endophytes, previous studies have proven fungal endophytes as the most reasonable modes for antibiotic discovery due to their cost effectiveness and environmental friendly nature. This study is based on the fungal endophytes from Acrostichum aureum, an associate mangrove fern. By nature, mangroves are hotspots for fungal endophytes due to their exposure to unfavorable environmental conditions. In turn to the nutrients obtained, endophytes generate various secondary metabolites and help mangroves to fight the pathogens and to withstand those harsh conditions. As an abundant fern, Acrostichum aureum plants are being used for consumption and various domestic and medicinal purposes. Similar studies conducted on them have also generated good outcomes. Thus, they were chosen for this study. General objective of this research is to determine the antibacterial activity of mangrove fungal endophytes associated with Acrostichum aureum. Others are to isolate and identify fungal endophytes from the chosen plant and to supply additional data regarding the subject. This is the methodology used in this study. Initially, roots, stem and leaves of Acrostichum aureum were collected 48 hours prior to the research. The samples were subsequently washed with sodium hypochlorite, ethanol and distilled water for surface sterilization. Then, the samples were cut into smaller pieces, plated on potato dextrose agar media and incubated at 25 degrees Celsius for 5 days. Then they were subcultured on potato dextrose broth to obtain pure isolates. The distinct colonies were examined using light microscope. The morphological and microscopic features of the fungi were used to identify the respective species by comparing them to previous publications. The broth cultures were filtered and subjected to solvent extraction using ethyl acetate. The extracts were dissolved in dimethyl sulfoxide and stored at -4 degrees Celsius for antibiotic sensitivity tests such as well diffusion, minimum inhibitory concentration and minimum bactericidal concentration against Escherichia coli and Staphylococcus aureus. In these tests, gentamicin was used as the positive control, while autoclaved distilled water was used as the negative control. For well diffusion, wells were created on the Muller-Hinton agar plates, and then the crude extracts were deposited on them, following which the diameter of the zones of inhibitions were analyzed. Next, 6 different dilutions of the crude extracts were generated, which were incubated with the test organisms and checked for their turbidity to determine the minimum inhibitory concentration. For minimum bactericidal concentration selection, the dilution corresponding to the MIC and those lower were swabbed onto nutrient media and were observed for the number of colony forming units. The results were collected and subjected to data analysis. As the result of fungal identification, 3 different fungal species, each per a part, were identified. Aspergillus fumigatus was identified from roots. As shown, it generated yellowish-white droplet-like colonies and possessed conidiophores ending with columnar-shaped heads. Aspergillus ochraceus was isolated from stem, whose colonies were pale yellow and the microscopic structures possessed globular conidial heads. Penicillium chrysogenum species were identified by the observation of brush-like conidiophores from leaves. As shown in figure 7, the positive control and the fungal crude extracts has diffused through the media and prevented the growth of the test organism, creating zones of inhibition. The diameter of these zones were measured and analyzed using the two-way ANOVA test, which were used to generate the graph in figure 6. As shown, all of the extracts except Aspergillus fumigatus against Staphylococcus aureus and the negative control were inhibitory. Failure to inhibit may be due to the Staphylococcus aureus' resistance against the metabolites of Aspergillus fumigatus. The highest inhibitory action was exhibited by Penicillium chrysogenum. Moving on, the lowest concentration, that is the highest dilution with zero visible colonies, was selected as the MIC. According to the results on table 1, zero visible colonies were observed from the third dilution and below. Thus, it was selected as the MIC against both the test organisms. It can also be observed that the number of colonies increased with the descending concentrations, suggesting the influence of concentration on the antimicrobial potency of the crude extracts. The lowest concentration with zero colony forming units was selected as the minimum bactericidal concentration of the crude extracts. As in table 2, MBC varied from extract to extract. Accordingly, Aspergillus ochraceus extracts possess the highest bactericidal activity against both the test organisms, while that of Aspergillus fumigatus was the lowest. Based on the results, we were able to conclude that all of the endophytic fungal crude extracts isolated from Acrostichum aureum possessed antibacterial actions against both Escherichia coli and Staphylococcus aureus. The highest inhibitory action was exhibited by Penicillium chrysogenum, while that of Aspergillus ochraceus was highly bactericidal. It was also observed that both MIC and MBC results varied from extract to extract. In order to improve this research, it can be recommended to perform molecular identification of the fungal species and to use spectrometric analysis to find the concentration of the crude extracts. The study can be expanded by comparing the results to those generated by more concentrated extracts. These are some of the references used in this study. Thank you.
Can ask any question from Ms. Kavanya related to this presentation.
I'm Kavanya Rajan, and this presentation is about
Yeah. Ms. Kavanya, what else? I know this is the only pteridophyte, the only plant seen in the mangrove habitat, right? So do you have any idea about whether this plant is already being used by the community for such purposes? In a different form like?
Good afternoon, madam. Am I audible? And
Can you hear me, Kavanya?
Good afternoon, madam.
Kavinya, can you hear me?
Yes, madam, I can hear you. Am I audible and is my video visible?
Yeah. Do you have any idea about this plant, whether the plant has already been used for such purposes, right? For antibacterial purposes in the like the village level, the ground level?
Yes, for the folklore medicinal purposes. In the folklore.
Are you there, Kavinya?
Yes, madam.
She's not there, no? But she's there. She's unmuted also. Can you hear me?
Yes, madam, I can.
I think she has a very low voice. You can hear, but can't clearly hear.
Did you hear my question?
Can you hear me now?
Now okay.
Yeah, please talk. Please talk.
In the folklore medicine, various parts of Acrostichum are being used as antibacterial, antioxidative, and antitussive agents. For example, leaf extracts as well as the rhizome extracts are being used for wound healing. Wound healing is basically based on the property of Acrostichum aureum to inhibit the bacterial or fungal growth.
No, true. That is what you presented. But my question is whether it is being used right now by the people at the very lower scale, not the extract scale. Do they use leaves, or this is a fern, very popular fern in the mangrove system, right? From the literature, have you heard anything like that?
Yes, in the village level, people are still using Acrostichum aureum for various medicinal purposes.
Yeah, people have been using it really right now. So then is there any particular reason why you choose these two types of bacteria like the Escherichia coli and the Streptococcus?
Although in general, Escherichia coli and Staphylococcus aureus are not very harmful, they do possess some pathogenic strains which are now being recorded as antimicrobial resistant species. So that's why I have chosen both of those as the test organisms in this study.
Yeah, right. Any more questions from the participants, please? Since there are no questions, let me thank you and the team for presenting your research in a way of this presentation for our sessions. Thank you, Kavin and the team. Let's move to the second presentation. Right?
Thank you.
Yeah, the second presentation, the ID 66, ID 31, I'm sorry, ID 31, molecular docking study reveals pinoresinol as a potential inhibitor for Nipah virus glycoprotein. Authors are T. Eknaligoda, O. Pereira, and H. Mudalige. The presenter would be T. Eknaligoda. Over to you.
Good day to you all. I am Tharudini Eknaligoda. How would you feel if I told you that a wildflower that you ignore every day has the potential to combat a deadly virus, and that this was analyzed and verified without even stepping outside? Well, I'd be quite excited. This is easily achievable through bioinformatic techniques. It is novel, innovative, green, efficient, and cost-effective. The technique utilized in this research was a type of molecular docking called protein-ligand docking, where computational simulations mimicked the natural activity of ligands and proteins, followed by the inhibition of the protein due to the binding of the ligand. Thereby, it was found out that the phytochemical pinoresinol of the wildflower Euphorbia hirta had the greatest ability to inhibit the deadly virus Nipah out of the samples utilized. These are the topics that will be deliberated today. Though not commonly heard, Nipah is a potential epidemic-causing virus that had its first outbreak in 1998 in Malaysia, followed by sporadic outbreaks in Bangladesh and India, with the most recent one being in 2021 in India. Nipah is a zoonotic pathogen belonging to the Paramyxoviridae family with a negatively charged RNA genome that encodes 6 functional proteins. Its natural reservoirs are fruit bats of the Pterophis genus, which are endemic to South and Southeast Asia. Primary zoonosis occurs through infected bacteria, and the transmission among the secondary hosts, such as humans, occur via contact and aerosols. Nipah primarily affect the respiratory system, followed by the progression into the nervous system, where many of the infected expires due to acute encephalitis and/or severe respiratory distress. Moreover, the absence of licensed treatment for Nipah has resulted in a fatality rate of 75%, and still, the treatment for outbreaks is limited to the handling of symptoms. But novel therapies such as antibodies, repurposing of FDA-approved drugs, research on discovering a vaccine, and potential ligands to be developed into drugs are conducted to be utilized in a possible outbreak. This research was carried out to identify potential phytochemicals that can inhibit Nipah, which are abundantly found in plants endemic to South Asia, thereby also promoting novel and green innovations. Autodoc 4.2.6 software was utilized for docking as it is a free, user-friendly graphical interface with many bioinformatic publication bases. Pymol, Chimera, LigPlot, and BioVia software were utilized for visualization, analysis, and validation. Primarily, NCBI PubChem database was used to acquire the 3D structures of the phytochemicals, and their druggability was analyzed by SwissAdMe. Phytochemicals were selected based on their endemicity to South Asia, general antiviral activity, and specific antiviral activity against Paramyxoviridae family. Phytophenolic compounds such as lignins and tannins derived from plants such as Euphorbia hirta that have depicted antiviral activity against the Paramyxoviridae family through clinical trials were chosen apart from other phytochemicals employed in the study. Nipah virus attachment glycoprotein was chosen as the protein receptor out of the 6 functional proteins of Nipah. It is a virulent factor that mediates the adhesion of virus to the host cell and pathogenicity. RCSB-PDB accommodates 4 structures of the Nipah virus attachment glycoprotein, from which PDB-ID2VSM was selected as it had the lowest X-ray diffraction resolution and the highest score of 86.1% for the most favored regions in the Ramachandran plot. 2VSM comprises chains A and B, from which chain A was utilized for docking. Chain B is the human cell surface receptor effrin B, which complexes with the Nipah virus attachment glycoprotein during host cell attachment. Docking against 2VSM was carried out separately for each phytochemical using the blind docking technique, which is done without prior knowledge on the active sites in 2VSM. Starting from step 5, for docking, all the structures were initially converted to the PDF format using OpenBarbell. Then 2VSM was loaded onto Autodoc and prepared for docking by removing all the water molecules, chain B, and heteroatoms, along with adding polar hydrogen atoms and Coleman charges, and merging the non-polar hydrogen atoms. Afterwards, a phytochemical was loaded and prepared by adding GM charges, merging non-polar hydrogens, and setting its toast off. These steps were followed to optimize the docking process. Post this, the grid box was generated to comprise the entire receptor, as the blind docking procedure was utilized in the study, and the population size at 300 and GA runs at 50 was set, followed by the execution of the AutoGrid and AutoDock programs consecutively. The above was validated by redocking the natural ligand IPA of 2VSM and analyzing the binding free energy, followed by the Ramachandran plot analysis of the 2 complexes. The conformations and orientations of each phytochemical complex generated were validated by superimposing the redocked IPA to its native crystal structure and calculating the RMSD value. Top 3 binding free energies were observed in pinoresinol, salonic acid, and syringaresinol, out of which pinoresinol and syringaresinol are ligands that have been proved by Lin et al. To be effective against Paramyxoviridae viruses. Docking in autodock is an iterative process where the population size or the collection points in space and the number of GA runs or the number of iterations define the quality and reliability of the binding free energies of each output. Primarily, the binding free energy, which is the sum of final intermolecular energy, van der Waals forces, hydrogen interactions, dissolution energy, final internal energy, and torsional free energy, were employed in the selection of the best performing ligands against 2VSM, along with the ADME properties of the phytochemicals. Here, the principal limitation was the inability of the scoring function to tabulate accurate binding free energies, as halogen and guanidine-arginine interactions are not considered even though they show significances in regulating the protein-ligand interactions. The number of hydrogen interactions were decided from the structure of the ligand, and therefore ligand specificity to the protein is measured using hydrogen interactions, whereas hydrophobic interactions are defined by the binding site residues, and thereby an increased number portrayed efficient binding of the ligand to the protein. Pinoresinol demonstrated blood-brain barrier permeance, which is an effective pharmacokinetic property for treating febrile encephalitis condition associated with MIFA. The presence of common amino acid interactions between the redocked complex and native 2VSM concludes a well-performed redock and validation of the docking protocols. The orientation and conformation of the best redocked pores was validated as the RMSD values obtained following superimposition were less than 2 angstrom. In conclusion, pinoresinol was the most suitable ligand for a potential drug molecule against the Nipah virus attachment glycoprotein. Further in vivo and in vitro testing can be carried out for verification. Common amino acid interactions mentioned here are identified as novel binding site residues of 2VSM. Molecular dynamic simulations, which analyze the movements of molecules and atoms within a given span of time, that validate the results through the RMSD and RMSF values, should be carried out. The off-target interactions of each component should be studied using protein-protein interaction network functional enrichment analysis to understand how each component might interact with a simulated environment such as a cell. Pinoresinol and syringaresinol from Euphorbia hirta can be extracted using the crude aqueous method followed by supercritical fluid chromatography, which can be utilized as leads for antiviral development in wet lab procedures, and the generated Ki values can be employed as references in trials. Moreover, to discover universal therapeutic drugs against MIFA, molecular docking and dynamic studies should be conducted using this study samples by using other functional proteins of MIFA. I conclude this presentation. Thank you.
Yeah, thank you very much, Miss Eknaligoda. You're very informative and a good research presentation, right? So now the floor is open for discussion. Any questions from the audience from Miss Eknaligoda? Yeah, it's maybe an ongoing research. It's a very nice presentation based on very nice research based on bioinformatics, really what is needed by the country right now. And I have one simple question. Is this pinoresinol is easily available? Are there any other plants which are capable of giving this kind of the material?
Yes, there definitely is. The lignins are what we're looking for. So pinoresinol is a lignin. So
Yeah.
pinoresinol has been found in other plants as well, such as I can't specifically remember, but there are plants that the principle is that the lignins are what actually works against maple.
Yeah, I heard the olive oil is rich with this particular compound, as I heard.
Mm.
Yeah, maybe you can see more literature in the future.
Yes. Yeah.
And any additives did you use for ligand proper binding?
එවලා තියෙනවා. අදම ඕනද?
Did you hear me?
No.
Are you using any more additives like for proper to facilitate the binding?
Just the Coleman charges and the GM charges are what we used for this research because Autodoc only facilitates that in particular. But if we use another software, we can actually really specify and go into using a specific additive, such as other charges, something much more stable. But Autodoc only facilitates these two charges.
Yeah, right. Thank you so much. It's a very good piece of research work. I hope the participants would gain sort of knowledge along that line. Thank you very much, Ms. Eknaligoda and the team.
Right?
Thank
you very much.
So let me move to the third presentation in today's parallel session number 3, the biological sciences, right? That is ID number 66, right? Changes in fast food consumption and marketing during the COVID-19 pandemic in the Kegol district, Sri Lanka. The authors are LDVS Gunawardana, AMMU Adhikari. So the presenter would be LDVS Gunawardana. Over to you, Mrs. Gunawardana.
Good afternoon, all of you. Welcome to our research presentation regarding changes in the fast food consumption and marketing during the COVID-19 pandemic in the Kegol district, Sri Lanka. Fast food is a quick, easily accessible, inexpensive alternative to home-cooked meals. Sri Lanka and most of the countries were employed COVID-19 pandemic with lockdowns to restrict the virus transmission. It was significantly impacted on millions of people's food consumption patterns. Restaurant industry had been particularly profound, with many establishments battling to maintain their economies while restaurants closed for sit-down service. Several countries were conducted research to determine the impact of the COVID-19 pandemic on general food consumption and purchasing behavior, snacking habits, and impact on fast food consumption. In Sri Lanka, just a few research had been carried out to see the effects of COVID-19 on food industry and food consumption. So the major objective of this research to investigate the changes in fast food consumption and marketing during the COVID-19 pandemic in the Kegol district. A survey-based study was conducted using 2 questionnaires, with one self-administered online questionnaire for consumers and another interview-administered questionnaire for fast food marketers throughout the Kegol district. The sample sizes for the customers and sellers surveys were conveniently selected. The customer-based survey was designed to yield a nationally representative sample of participants, and it was aimed at 250 participants, out of which 201 responses were received. For the sellers-based survey, 39 participants were responded from the expected 50 participants. Both questionnaires were conducted in the English language and translated to the Sinhala language. Data was collected from 8 November to 10 December 2021. Coding and entering of the gathered data was done using Microsoft Excel, and the statistical analysis was performed using SPSS version 16 software. According to the results of the consumer survey, based on the comparison between pre and during the COVID-19 pandemic situation, the consumption frequency was significantly decreased. The number of the frequent consumers, which means daily or 3 or more days per week consumers, were reduced by 14%, and the number of the moderate consumers, which means 2 or 3 days per week or 1 day per week consumers, were also reduced by 9.4%. And the participants who consume fast food rarely was increased by 21.4%. The weekly expenditure for the fast food also significantly decreased. Expenditure between 500 rupees to 1000 was reduced by nearly 40%, and the expenditure between 1000 rupees to 3000 rupees was reduced by nearly 31%, and the expenditure less than 100 rupees was increased by nearly 76%. And the preference for online ordering and preference for mobile delivery services were significantly increased. Before the COVID-19 situation, 14% were preferred to online orderings, but during the pandemic situation, it was increased to 28%. As well as for mobile delivery services, before the pandemic situation, 30% were preferred to purchase fast food items, but during the pandemic situation, it was increased to 64%. Also, according to the chi-square test, to investigate the associations between consumers' demographic variables with the fast food consumption changes, it was revealed that there were positive associations between preference for online ordering during the COVID-19 period with occupation and monthly income, and also for the reduction of the consumption. Travel restrictions and lockdown situations were noted as major reasons. As the consumer results, the comparison between pre and during the pandemic situation for the marketers survey, the number of the daily customers was significantly decreased. Based on the details of the participants, customers between 100 to 300 was reduced by 93%, and the customers more than 300 reported as zero during the pandemic situation. Customers less than 10 was increased by 32%. Daily revenue also significantly decreased during the pandemic situation. The daily revenue more than 50,000 rupees was reported as zero respondents. The daily revenue between 10,000 rupees to 50,000 rupees was also reduced by 68%, and the daily revenue less than 2,000 rupees was increased by 27%. And also, marketing at the shop was significantly decreased. Nearly 21% of the shops was closed during the peak of the COVID-19 situation in Sri Lanka. Marketing through online orderings was significantly increased. Before the COVID-19 situation, 24% marketers provide online marketing system, but during the pandemic situation, it was increased to 40%. Mobile delivery services also slightly increased, but it was not shown any significant difference. During the pandemic situation, nearly 24% of the marketers were introduced newly invented fast food recipes, while 22% offering takeouts or delivery services efficiently as their strategies for consistent marketing to reduce the huge drop of the marketing of fast food items during the COVID-19 situation. As the overall results of the both consumer-based survey and fast food marketing survey, 59.7% of the consumers were reported that the reduction of the consumption. For the reduction, nearly 96% said that the travel restrictions and lockdown conditions, and 82% said that the consumption of fast food items were not suitable for their health conditions because of isolating at home for a longer time period as their major reasons. 67% said that it was too heavy for their budget balance as their reason for the reduction of the consumption. 19.9% of the consumers were reported that the increment of the consumption as their major reasons. 90% said during the lockdown period, entire family at home and the children like to fast food items more than home prepared meals. 20.4% of consumers reported that no any change of their consumption. According to the results of the marketers, 89.75% reported that the reduction of the marketing. 100% of the marketers agreed with the travel restrictions as the major reason for the reduction of the marketing, and nearly 50% said that the lower of the production due to the lack of the employees caused for the reduction of the marketing. 7.7% were reported an increment of the marketing and reported that online ordering and mobile food delivery services were going successful as their reason for the increment. 2.6% of the marketers reported that not any change of the marketing. As the conclusions of this study, consumer fast food consumption behavior has changed due to the COVID-19 pandemic. The frequency of fast food consumption and its expenditure has significantly decreased, while online ordering and purchasing from mobile food delivery services were significantly increased. Also, there were positive associations found between preferences for online ordering during the COVID-19 pandemic period with occupation and monthly income of the consumers. Fast food marketing behavior has also changed due to the COVID-19 pandemic situation. The number of the customers and daily revenue has significantly decreased, while online ordering was significantly increased and the delivery services have also increased. The fast food marketers have taken measures to increase their revenues and to ensure customer and food safety during the pandemic period. As further suggestions, need to identify fast food and other general food consumption and purchasing behavior during the COVID-19 pandemic situation for the whole population with various demographic, socio-economic and educational backgrounds in Sri Lanka, and also need to identify the fast food marketing behavioral changes for the whole population during the COVID-19 pandemic situation in Sri Lanka. And these are some references regarding this study, and this is the end of my presentation, and thank you all of you.
Thank you very much, Ms. Gunawardana. Your presentation related to very recent incident, the COVID-19 pandemic. And now the floor is open for questions, right? Please ask questions when you feel relevant. From Ms. Gunawardana. In the absence of
Yeah,
questions,
can I just,
yeah, sure.
I was just wondering, the study is based on changes in fast food consumption. So when you were looking at collecting the data, were you, did you have data for pre-COVID time or post-COVID time, or did you just ask in the questionnaire itself how their behaviors changed?
Ms. Gunawardana, did you hear the question? Ms. Gunawardana, can you hear us? I'm sorry, The Gaya person is not available.
That's okay. That's all right.
Yeah. Yeah, so let's move to the next presentation. Thank you for the informative presentation by Ms. Gunawardana. Let's move to the other presentation. Next one, the ID 103. Primary malaria vector Anopheles cruciparis gives abundance and climatic correlation in a post-malaria elimination setting. The authors are HMP Heva Vitarana, G.R. Hanavaka, MDJ Sapramadu, R.G. Premaratna, H.T.R. Jayasurya. The presenter would be HMP Heva Vitarana. Over to you.
Hello everyone. Our topic is primary malaria vector Anopheles caulicifacis guides for abundance and climatic correlations in a post-malaria elimination setting. Malaria is a vector-borne disease and it was a major public health problem in the past and it was eliminated from Sri Lanka in 2012. After malaria elimination, there is a high risk of re-establishment of malaria to the country, mainly due to increased importation risk and increased receptivity. Importation risk and the receptivity are the two major factors contributing to malariogenic potential. And in Sri Lanka, at the moment, importation risk for malaria is due to the transfer of human to and from neighboring malaria endemic countries. And receptivity for malaria is mainly due to the abundant presence of Anopheles vector mosquitoes and ecological and climatic factors favorable for malaria transmission. And in Sri Lanka, of the malaria vectors, Anopheles caulicifacis giles is considered as the primary malaria vector. And in the past, major factor that contributed to the distribution of malaria in Sri Lanka was the favorable climatic conditions for Anopheles caulicifacis prevalence in different geographic areas. Mainly, Sri Lanka is divided into 3 geoclimatic zones, namely wet zone, intermediate zone, and dry zone. And it was noted that in the past, malaria was endemic in the dry and the intermediate zones, and it was epidemic in the wet zone. And there are various previous studies that has revealed the abundant presence of Anopheles caulicifacis in the 2 endemic zones, but there is very limited information of Anopheles caulicifacis distribution in wet zone. And another factor that we highlight is that in the wet zone, at the moment, there is a high risk of malaria reintroduction due to the high transfer of rate of labor within malaria endemic countries. This is specific for Western province situated in the wet zone. And the findings of this study was expected to be useful in determining the factors contributing to receptivity in the selected study sites and thereby contribute to decision making for sustaining the malaria eliminated status in Sri Lanka. There were 2 objectives of the study, and first one was to assess the distribution and abundance of Anopheles caulicifacis across all climatic zones of Sri Lanka, and secondly to correlate with climatic factors, rainfall, temperature, and relative humidity with Anopheles caulicifacis densities. The study was carried out in 5 study sites representing the 3 climatic zones of Sri Lanka, and of 5 study sites, 3 were located in the dry zone. And the first one was in Horopatan MOH of Anuradhapura district, and the second study site of dry zone was in Punakari MOH of Kilinochchi district, and the third study site of dry zone was in Buttala MOH of Monragala district. And the other 2 sites were the Nikavarati MOH of Kurunegala district representing intermediate zone, and Mirikama MOH of Gampaha district representing wet zone. In each of the above 5 study sites, 2 GN areas were selected based on the historical evidence of malaria transmission. And the climatic data of the 5 study sites were collected from the Department of Meteorology, Colombo. And it was on monthly basis. Anopheles caulicifacis mosquitoes were collected on monthly basis from April 2015 to March 2017 during 24 surveys. And there were 4 sampling methods used for collection, and out of these 4, 3 were adult collection methods, namely cattle baited trap collection, CBTC, human landing catches, HLC, and Indo-Han collection, IHC. For the larval surveys, dipping technique was used to collect the immature stages of Anopheles culicifacius, and all collected adults and larval specimens were identified for the species level on external morphology using standard identification keys. Data analysis. Mean densities for each adult trapping method was calculated by dividing the number of Anopheles culicifacius mosquitoes from the total numbers and sampling efforts per month. For CBTC, the mean density was calculated as number of females per trap. For Indo-Han collections and human landing catches, mean densities were calculated as number of females per man-hour. And for the immatures, the larval surveys, mean density was calculated as number of larvae per 100 dips. Then, multiple comparisons of the mean densities were done. Kruskal-Wallis test was used for this purpose with Dunn's post-hoc test for multiple comparisons, and it was done using SPSS 21 package. And for each study site, mean monthly climatic variables were correlated with the mean Anopheles culicifacius densities using Spearman's rank order correlation matrices using the PAST software. And these two statistical techniques were non-parametric tests. Results. A total of 10,381 Anopheles culicifacius adults and larvae were collected by the 4 collection methods during the 24 surveys. And it was noted that the highest number of culicifacius specimens were collected from the Nicovareti MOH located in intermediate zone. In the Nicovareti site, the abundance of percentage for CBTC was 74.2 and it was 52.7% for human landing catch and 88% for Indo-Han collection and 63.1% for larval surveys. And the second highest abundance of culicifacius was noted from Puttala site located in the dry zone. And it was observed that in the dry zone Poonakari site, the lowest abundance of culicifacius was seen from CBTC and LS. This slide shows the distribution and abundance of Anopheles culicifacius in cattle-baited trap collection and larval surveys in different study sites. Densities of culicifacius was not similar between the 5 study sites based on the Kruskal-Wallis test for the 2 techniques. For CBTC, its statistic was 43.2 and the p-value was below 0.001. And the Dunn's post-hoc test showed that densities from CBTC in Nicovareti were significantly different from the Horopatana, Poonakari and Medigama sites. And for the larval densities, its statistic was 15.4 with a p-value below 0.01. And again, Dunn's post-hoc test showed that the larval density of Nicovareti was significantly different from Poonakari and Medigama sites. This slide shows the distribution and abundance of Anopheles culicifacius caught from human landing catches and Indo-Han collections in the different study sites. Again, the densities of Anopheles culicifacius were not similar between the 5 study sites from these 2 techniques based on Kruskal-Wallis test statistic. For human landing catch, its statistic was 45.2 with a p-value below 0.001. And the Dunn's post-hoc test showed that the density from human landing catch in Nicovareti was significantly different from the densities of Horopatana, Poonakari and Medigama sites. And for Indo-Han collection, its statistic was 24.4 with a p-value below 0.001. And again, Dunn's post-hoc test showed that the Indo-Han collection density of Nicovareti was significantly different from the other 4 sites. This slide shows the correlations of mean densities of Anopheles culicifacius from 4 collection techniques with the 7 parameters. And in this table, it is given only the significant correlations at p-value below 0.05 in blue color and 0.01 in red color. The climatic parameters correlated were current month rainfall as lag 0, previous month rainfall as lag 1, 2 months back rainfall as lag 2, and maximum temperature, minimum temperature, average temperature, and relative humidity. It was observed that adult densities from indoor hand collection and human landing catches in Mirigama had a negative correlation with current month's rainfall. Again, larval densities of Mirigama had a strong negative correlation with the current month rainfall. Moreover, larval densities of Nicovarity also had a negative correlation with 2 months lag rainfall. And temperature and Anopheles calyphysis density showed a significant negative correlation in Horopatana from CBTC with average temperature. And again in Buttala, human landing catch and indoor hand collection densities with maximum temperature. And in Nicovarity, human landing catch and larval service with average temperature. Finally, in wet zone Mirigama, Anopheles calyphysis densities from CBTC showed a negative correlation with minimum temperature and average temperature. And also human landing catch, indoor hand collection and larval densities showed a negative correlation in Mirigama site with minimum temperature. Therefore, based on the findings of this study, it was understood that Anopheles calyphysis is distributed in all climatic zones with a spatial variability for its abundance, specifically in breeding, biting and indoor resting behavior. And compared to the 5 study sites in Nicovarity, intermediate zone was prominent for calyphysis, breeding, biting and indoor resting populations. And even within the dry zone, there were differences among study sites in distribution and abundance. For example, in the Punakari site had the lowest densities of calyphysis and there were no human biting or indoor resting populations. And in Mirigama, in wet zone site had high abundance of human biting populations when compared to the dry zone Horopatana site. The second conclusion is that rainfall, temperature and relative humidity play a role in distribution and abundance of calyphysis within the climatic zones. And it was seen all climatic factors had negative impact on breeding, biting and resting populations in all study sites. Specifically, it was observed that characteristic climatic factors of the wet zone do not appear to be favorable for vector-host contact under normal weather patterns. In the intermediate zone, lack of rainfall in previous months would be the reason for high abundance of the larval density. Therefore, in the future efforts for determining malaria receptivity, we suggest to elicit the importance of climatic correlations with vector abundance. Finally, authors acknowledge financial assistance from the Open University of Sri Lanka and Anti-Malaria Campaign for data collection and all the field staff of Anti-Malaria Campaign for their assistance for field collections. Thank you.
Yeah, thank you very much, Ms. Heva Vitaran and the team for the wonderful presentation, right? So let's open the presentation for discussion. Any questions? Yeah, it's very informative presentation really you have done in the setting of the malaria, about the malaria vector. As you said very clearly, is there any reason like from the literature, either based on social background or any other particular situation related to this particular area, the Nicovarity for high abundance? Just for my curiosity, I'm just thought of checking with you whether you either through literature or just from the social context.
Actually, all the study sites were selected based on the previous malaria history, the incidence history, and particularly out of these 5 sites, 4 sites had major river systems. So in Nicovarity, the major river system we had was Daduroya. So mainly related to the primary vector, malaria vector, calyphysis, breeding habit, they usually prefer to breed in river systems, in the drying up river beds. So mainly due to the climatic factors prevailing in equality and based on the availability of the breeding sites, we suggest that there was high prevalence in equality side.
Yeah, right. Maybe true. Yeah, right. So the same question from me regarding the Mirigam site as well. The whiting is more like whether it is any particular reason like that in the
Yeah,
Mirigam
actually,
site.
in the biting, you were asking about biting?
Yeah, yeah. As the data you indicated, the whiting is more in the Mirigam site to somewhere, I don't know, I may be wrong. Somewhere you mentioned the Mirigam, some sort of like high, something special related to Mirigam area mentioned somewhere.
Yeah, the biting was the third highest in Mirigam, and the indoor resting was the second highest.
Yeah.
So those are the two adult parameters that we check for malaria transmission potential. So as this study was conducted during a 24 months period, that was due to the seasonality, because in Mirigam sites in the wet zone, it was indicated that the adverse, not the normal weather patterns, but when the rainfall is in shortage and when the temperature was not the same, that favors for the adult vector survival. So those two parameters goes with the survival of the adult vectors.
Okay. Thank you very much, Ms. Mrs. Hemavitaran and the team for the very informative presentation, right? Thank you so much. So let's move into the next presentation, right? That is ID 107, medicinal properties of Banga Eclipta alba hasap from the perspective of Unani medicine, a review. The authors are MRF Prifna and MAF Busra. The presenter would be MRF Prifna. Over to you.
Good morning, all of you. I am Rifna from Institute of Indigenous Medicine, University of Colombo. It gives me an immense pleasure in presenting the review on medicinal properties of Bhangra Eclipta alba from the perspective of Unani medicine. This is the outline of my presentation. Unani system of medicine comprised 4 different modes of treatment, such as pharmacotherapy, dietotherapy, regimental therapy, and surgical therapy. Out of this, pharmacotherapy plays a major role, which mainly includes the use of herbs for the treatment modalities. Although many of the herbs were subjected to clinical studies, there are still certain herbs which require further studies on therapeutic effects. On that basis, Eclipta alba, known as Bhangra in Unani system of medicine, is an abundantly available but underutilized herb that is commonly grown as a weed without any much intervention. Eclipta alba, known as Bhangra, belongs to the family Asteraceae, genus Eclipta, and the species Alba. Thus, this study was done with the main objective to systematically collate the data of Eclipta alba in Unani perspective, and with 2 other specific objectives, that is to ascertain new study proposals on therapeutic uses and pharmacological actions on Eclipta alba from Unani perspective, and also to probe the Unani herbs among the exotic plants of Sri Lanka. As we know that many of the herbs that are mentioned in Unani classical texts are not available in Sri Lanka. So whatever the herbs that are mentioned can be probed among the exotic plants. This study was carried on the basis of a systematic examination of classical Unani literature. For that, 4 main classical Unani texts were used, such as Maghzanul Mufradat, Manafiul Mufradat, Unani Adya Mufrad, and Kitabul Mufradat, along with research articles entitled on pharmacological effects and therapeutic effects, which were abstracted from index journals from 2012 to 2020 through search engines such as PubMed, Mendeley, and Science Direct. The research articles related to commercial and agriculture were excluded. Bhangra, Aclipta alba, pharmacological actions, and therapeutic effects were the key words used in the search engines. So this is the results that we have found from the Unani Classicals. Unani Classical mentions Bhangra as an annual herb that grows up to a handful tall or sometimes even shorter. The stem of the plant is mostly fleshy and strigous with scanty leaves that are much similar to the broad basil leaves. The leaves of Bhangra are simple, lanceolate to oblong in shape, tapering to the base with scanty adpressed hair on its either sides. The leaves are arranged opposite, they are stipulate and sessile. Flowers of Bhangra are numerous in number, they are white in color, bisexual, calyx reduced to scantily hairy teeth, and petals are 4, which is fused into a tube and expanded distally. The fruit of the plant is oblong in shape, they are compressed, sides are covered with warty excellence, sometimes with minute spines. The seeds of Bhangra are dark brown in color and hairy. Roots are also well developed. According to the temperamental theory of Unani medicine, this plant belongs to hot and dry temperament. And there are 3 varieties mentioned in Unani Classicals: yellow, white, and black. White variety is used in many diseases. That is, the white one has lot of pharmacological actions. So they are used medicinally. The black variety is mainly used only as a hair growth promoter and it is used to blacken the hair. And the yellow one has the least therapeutic effects and they are not used medicinally. The entire plant of Bhangra is used medicinally. Alkaloids, acliptin, and nicotine are abundantly present. The principal constituents of Aclipta alba are cosmostern derivatives, vadolactone. Other constituents such as acliptal, hepatocosanol, and stigmasterol are also present. There are several actions mentioned in Unani Classicals. That is, it acts as a blood purifier, aphrodisiac, hepatoprotective action. It is a very good cholagogue, anti-inflammatory, eye tonic, and carminative, and as well as it is a very good deobstruent. Based on these pharmacological actions, Bhangra is used to treat various diseases. The entire plant of Bhangra is used in chronic skin diseases and also to treat syphilis. With castor oil, it is given in worm infestation. And also this plant is used as a ear drop in earache. The distillate of the leaves of the plant are used as eye drops in conjunctivitis. The juice of the plant is used in jaundice and also in uterine hemorrhage. In scorpion stings and serpent bite, the leaves of the plant are used both internally and externally. The decoction made out of the whole plant is given in hemorrhoids and kata. Further, the leaves are applied externally to relieve headache. They are applied on forehead to relieve the headache. And Bhangra is incorporated into Unani compound formulation as an ingredient, such as Majune Bhangra and Rogane Bhangra. Rogane Bhangra is an oil that is beneficial as a hair growth promoter, and it is used externally in skin diseases. Majune Bhangra is a semi-solid compound formulation that is used internally to treat liver diseases, uterine hemorrhage, catarrh, and hemorrhoids. These are some pharmacological actions that are suggested by the studies. Hepatoprotective action, anti-epileptic, anti-cancer, analgesic, anti-hemorrhagic, promoter for blackening and growth of hair, neuroprotective, anti-Parkinsonism, antioxidant, anti-arthritic, antibacterial, and antifungal. Several studies elicit that hepatoprotective action of Eclipta alba, which can be correlated with Mukaviya jigger and Muwallide safra, which has proposed in Unani classical text. Few more studies suggest that neuroprotective, anti-epileptic, and anti-Parkinsonism actions that can be correlated with Mufatite Sudat and Muhallile Varan as per the Unani view. Further, Mustafiyahun Muhallile Varam action are much more relevant to anti-carcinogenic effects. So, this plant, Eclipta alba, which is commonly grown as a weed, possesses wide range of therapeutic effects, and it has been used to treat various diseases since ancient times. Further, recent clinical studies depicted that it possesses hepatoprotective and anti-inflammatory actions, which are much relevant to the actions mentioned in the Unani classical text. Suggestion what I can recommend or suggest is that detailed clinical studies on aphrodisiac and eye tonic. These aphrodisiac and eye tonic actions are mentioned in the Unani classical text, and in this action has to be explored. These are the references, and thank you.
My studies topic is prevalence and bioenergy.
Thank you very much, Ms. Rifna, for the presentation done with much data. Let's open it for discussion. Any questions from the audience?
Can I ask this
Sure.
question?
Yeah. You can ask, can you hear me? Yes, you can ask. Ms. Rifna, are you here?
Yeah, madam, I'm here.
That's really, yes. Can you please let me know?
Madam, question is not audible. Can you please repeat it? I didn't hear.
If you're referring to me, it is, I just want to find out what Bhangra in Sinhala is.
It's known as Bhingraj in Sanskrit.
Sorry, Banga.
Ayurveda
in
Singraj.
Isn't it
Singaraja.
Kirindia?
Kikirindiya in Sinhala.
Yeah. That is what
Yeah.
the audience is asking, the Sinhala. That is
Okay.
Kikirindia.
Kinkirdia?
Kikirindia.
Kikirindiya.
Kikir. Okay. Thank you.
Right. Since any more questions from the audience? Yeah. Can I just suggest you a small improvement for your review, Ms. Rifna? Yeah, you could have linked now you have said in the text like the certain compounds that are available in this plant, right?
Yeah.
So you could link that with the whatever the impact you are saying, like the blood purifier or anything like that. So you could, if you add that compound, that the metabolic process or the how it is done or something like that, may be useful for the reader so that they will get to know like the, rather than having a general term. So there would be some sort of use for them, like for the readers.
Okay.
You
Thank
have
you.
already started, you may be doing it later, right? So with that, it
Yeah.
will be a good review, right? Okay, thank you very much, Ms. Rifna. Since there are no more questions, so let me move to the next
Thank you.
presentation, right? Next presentation, the ID 153. It will be on prevalence and bionomics of the Anopheles mosquitoes in selected sites in the Hambantota district. Authors are S. Priyadarshani, G.R. Ranawatha, P.J. Jude, H.T.R. Jayasurya. The presenter will be S. Priyadarshani. Over to you, Ms. Priyadarshani.
My studies topic is prevalence and bionomics of the Anopheles mosquitoes in selected sites in the Hambantota district. This presentation is organized in the order of introduction, objectives, methodology, data analysis, results, conclusion, and acknowledgement. Moving to introduction. Malaria is considered as a life-threatening disease. It was one of a major public health problems in Sri Lanka. The last indigenous malaria case was reported in 2012. After successful completion of 3 years with 0 indigenous malaria cases, the country was certified by the World Health Organization as a malaria-free country. Currently, Sri Lanka is in the phase of prevention of re-establishment of malaria. Keeping malaria-free status is a challenge due to continuous influx of imported malaria patients from malaria endemic countries and high abundance of malaria vectors in many parts of the country. Hence, it is very important to understand the current status of vector prevalence and their bionomics in vulnerable areas in order to take appropriate vector control decisions. Anopheles culissipesis is considered as the primary vector of malaria in Sri Lanka. Other potential vectors are Anopheles subpictus, Anopheles varuna, Anopheles annularis, Anopheles vagus, Anopheles tessellatus, and Anopheles stephensii. When we consider the Hambantota district, it is highly vulnerable for malaria with the presence of harbour, airport, and many tourist attractions. With this importation risk of malaria, it is crucial to understand the vector bionomics in the district, but there are no published evidence on bionomics of Anopheles vectors in Hambantota districts. Hence, the aim of this study was to examine the prevalence and bionomics of Anopheles in the Hambantota district. There were 3 specific objectives of this study. First is to study the prevalence, species richness, diversity and relative abundance of larvae and adult of Anopheles vectors, to determine the breeding habitats of Anopheles vectors, and to determine the biting behavior of Anopheles vectors. 4 study sites were selected in Hambantota district, Arabukka, Mirichavila and Harbour localities in Hambantota MOH, Mattala Airport site in Lunugambehera MOH. These study sites were selected based on previous high malaria endemicity and high malaria receptivity and vulnerability. Monthly mosquito sampling was carried out from October 2021 to March 2022. Larvae were collected using larval surveys. Adults were collected using cattle-baited trap collection and cattle-baited herd collection. Both larvae and adult were identified using morphological identification keys to species level. Secondary data of outdoor human landing night collections were obtained from anti-malaria campaign headquarters, unable to perform indoor techniques such as indoor human landing night collection and indoor hand collection due to COVID-19 pandemic. Data analysis. Species abundance in study sites was compared using one-way ANOVA using SPSS. Species richness and diversity in study sites were analyzed using Shannon-Weiner Diversity Index. Larval densities were calculated as density per 100 days. Adult mosquito densities were calculated as per trap and per hut densities. Moving to results section, prevalence of Anopheles in Hambantota district. A total of 7,072 mosquitoes comprising 829 larvae and 6,243 adults belonging to 11 Anopheles species were recorded from both Hambantota and Lulugambehera MOH areas. These were included with the primary vector and other potential vectors. Anopheles subvectus was the most predominant species in all techniques, representing 35% of the total Anopheline collection. Anopheles species composition. 10 species were commonly found in both Hambantota and Lulugambehera MOH areas. Those were Anopheles culissifacis, Anopheles subpictus, Anopheles nigerimus, Anopheles varuna, Anopheles vagus, Anopheles pediceniatus, Anopheles pallidus, Anopheles densai, Anopheles tessellatus, and Anopheles barbirostris. Anopheles annularis, the potential vector found only in Hambantota MOH area. Anopheles species richness and diversity. When considered the larvae, the highest species richness and diversity were recorded in Matsala study site. The highest species richness of Anopheles adults, 11, was recorded in Araboka and Mirichavila study sites. The highest diversity of adults, 1.86, was recorded in Matsala study site. The highest relative abundance of Anopheles culissifacis larvae and adult were reported in Mattala study site. The highest relative abundance of most predominant vector Anopheles subpictus larvae was reported in Mirichavila and adult were reported in Araboka localities in Hambantota MOH area. Anopheles nigerimus also found in high numbers in both MOH areas. This difference in diversity and relative abundance of Anopheles species may due to ecological and climatic factors favoring the larval development of some species. Further, comprehensive information on malaria vector species richness, diversity, and abundance in vulnerable localities is an important tool in planning and implementing successful vector control programs. Breeding habitats of Anopheles. 18 types of Anopheles breeding habitats were identified in both MOH areas under 3 categories: permanent breeding habitats, semi-permanent breeding habitats, and temporary breeding habitats. The major breeding habitats identified were canal, lagoon, tank, and ground pools. Human biting behavior of Anopheles mosquitoes. In Hambantota MOH area, Anopheles culissifacis, Anopheles subpictus, and Anopheles nigerimus were shown outdoor human biting behavior. The highest biting preference was shown by Anopheles subpictus, which is 87%. Relative biting preference of Anopheles culissifacis was 11%. Outdoor biting peak for Anopheles subpictus was observed from 19 to 21 hours. Anopheles culissifacis biting was observed during 20 to 22 hours. In Lulugambehera MOH area, Anopheles culissifacis and Anopheles subpictus were shown human biting behavior. The highest relative biting preference, 69%, was shown by Anopheles culissifacis. The relative biting preference of Anopheles subpictus was 31%. Outdoor biting peak of Anopheles culissifacis was observed during 19 to 21 hours. The peak biting hours of Anopheles subpictus was observed 18 to 20 hours. Conclusions. Both Hambantota and Lunugambera MOH areas are highly receptive to malaria with the availability of malaria vectors, breeding habitats, and the human biting behavior of Anopheles culissifacis and Anopheles subpictus, the primary and potential vectors of malaria in Sri Lanka. Anopheles subpictus was the most predominant species during the study period from all techniques, representing 35% of the total Anopheline collection. Anopheles culissifacis was the most predominant Anopheles species found in Mattala study site, which is in Lunugambera MOH area. It is recommended to continue the study to a period of 1 year in order to observe seasonal variations of Anopheles vectors. These findings will contribute towards the ongoing vector control activities being implemented in Hambantota district. I would like to acknowledge the Ministry of Health for financial assistance, entomology teams of Anti-Malaria Campaign Headquarters and Hambantota Regional Office for assisting field entomology service, Regional Director of Health Services, Regional Malaria Officer, Director Anti-Malaria Campaign for administrative and logistic support. Thank you very much.
Yeah, thank you, Ms. Priyadarshini, for the very informative presentation. Now the presentation is open for discussion. Any questions from the audience, from the participants? Seems no questions. Like, did you think about any particular reason, like for having such a big abundance in the Mattala area, like where the area being in the arid zone, in the climate wise?
Yes, in the Mattala, when compared to the Hambantota MOH, there were a lot of fresh water breeding habitats, such as lakes and stream margins. So those are very preferable sites for Anopheles vector breeding. So maybe for especially when it comes to Anopheles culissifacis, the main primary vector of malaria we found in high abundance in Mattala site because of the availability of preferred breeding sites in Mattala. That is mainly because of the availability of preferred breeding habitats there.
And this is a very general question actually for my curiosity, because usually in Colombo, the suburbs, like the mosquitoes are more abundant from 5 to 7, like as the way I have observed. But in your study, you have said that they are mostly abundant between 7 and 9 in the night, right?
Yes.
So any particular reason for that?
Yes, when we consider the Colombo district, we say Colombo is non-endemic for malaria. That means the Anopheles population is very less in Colombo. But Colombo, the dengue vectors, Aedes mosquitoes are highly prevalent in Colombo district because of the availability of their breeding habitats in Colombo. So actually in Colombo, we can, we say, dengue vectors bite in dusk and dawn. So after like 5 p.m., they get active. So mainly dengue vectors bite during that time, not Anopheles.
Not Anopheles, right?
Yes.
Okay. Thank you very much, Ms. Priyadarshini and the team for your excellent presentation and the research, right? It's a good piece of research. Thank you, right? So let's move to the next presentation. That is ID 156, biomonitoring of heavy metals in the yellow-billed bubbler in selected landscapes in Sri Lanka from a conservative perspective. Authors are TSP Fernando and F.S. Sully. The presenter would be F.S. Sully. Over to you, Sully.
Good afternoon, everybody. I'm F.S. Sali from the Department of Zoology, and I'm here to present our study on the topic biomonitoring of heavy metals in yellow-billed babblers in selected landscapes in Sri Lanka from a conservation perspective. Why is conservation important? So, in search of better opportunities for employment, healthcare, and education, people tend to move from rural to urban areas. We call this population shift as urbanization. Presently, around 56% of the world's population lives in urban areas. Urbanization, as good as it may sound, comes with its fair share of detrimental effects to the environment. And one such condition is the release of heavy metals. Over the years, these metals have accumulated in the environment, causing serious health effects. Therefore, there is a need to monitor them in order to conserve the environment. One commonly used technique to monitor heavy metals is biomonitoring, because biomonitoring provides first-hand information about deviations of normal patterns of biological response, and mainly it's cost-effective. Moreover, birds are very suitable organisms to monitor heavy metals as they occupy the higher levels of food chain. Taking all these things into consideration, Turdoides affinis, a common bird species, was selected as it is widely spread across the island. So our question for research was whether there is an effect to feathers or blood profile or aggressive behavior of birds residing in different landscapes selected based on the levels of urbanization. In order to find an answer to our research question, we carried out experiments around three specific objectives. Firstly, to determine the levels of heavy metals in feathers. Next was to study the blood profile of birds in relation to heavy metals from the three landscapes, and also to study the aggressive behavior of birds in relation to heavy metals from the three landscapes. To fulfill the objectives, three landscapes were selected based on the levels of urbanization. The premises of the Open University of Sri Lanka as the urban site, Horana in the Kaluthara district as the semi-urban site, and the Hiare Reservoir Sanctuary in Galle as the forest site. So a number of 6 birds were captured from each landscape using standard mist netting techniques. This capturing was carried for a period of 3 months, during which 2 nets were used, and the nets were opened twice during the day at different locations within the landscape. And the nets were checked regularly for birds. Upon the capture of birds, the parameters were recorded, the samples were collected, and thereafter the birds were banded and released. The morphometric characters of the bird was measured under the parameters mentioned here, such as the head length, the beak length, the tail length, and so on. And the plumage characteristics was measured based on the quality and extent of color around certain regions of the bird's body, such as the belly, rump, the primaries and secondaries, using spectrophotometric techniques. For the heavy metal analysis, 2 feathers were removed around the tail region from 3 birds in each landscape, and the collected samples were sent to the National Aquatic Resources Research and Development Agency. There, the feather samples were analyzed for the heavy metals, lead, mercury and cadmium, using the atomic absorption spectrophotometric technique. For the blood profile analysis, a volume of 100 microliters of blood was collected from the ulnar vein into microhematocrit tubes, and the collected sample was transported to the laboratory. In the laboratory, the blood was analyzed under the 6 parameters mentioned here, such as the blood cell counts, the hemoglobin concentrations, a differential count, and using the parameters, the blood indices such as mean corpuscular volume, the mean corpuscular hemoglobin, and the mean corpuscular hemoglobin concentration were calculated. Now, to study the aggressive behavior, a decoy owl and a speaker were set up in a few locations in the selected landscape. Then the call playback was played in the natural volume for about 2 minutes or until the birds recognized. So this technique is called the stimulation territorial intrusions. So based on the set aggression scores as mentioned on this slide here, the aggressive behavior was then determined, taking 0 to 2 as scores for non-aggressive behavior and 3 to 5 as scores for aggressive behavior. What we see here is the results obtained for heavy metal accumulation in feathers. It is clear that heavy metals were reported from all three landscapes, with the highest being reported in the natural landscape. In the morphometric analysis using PCA, there was no significant variation observed. This tells us that the bird's body and structure is not affected despite the presence of heavy metals. But in the plumage analysis using PCA, there is a significant variation shown by birds in the urban landscape. From this, it could be understood that heavy metals have an effect on the feather color of birds. From the results obtained the blood profile, it is clearly evident that there were striking deviations in blood parameters such as the differential count, the Paxil volume and hemoglobin concentration, and also to blood indices. And this could be related to the heavy metal accumulation in the three landscapes. And in the results obtained for aggressive behavior, the highest percentages of aggression were reported from the urban and semi-urban landscapes, and the lowest is reported from the forest landscape. The study concluded that heavy metals accumulate in all landscape irrespective of urbanization, and the highest levels were reported in the natural landscape. The reason for this is the presence of an agricultural land adjacent to the IOURS sanctuary. So the birds residing in the sanctuary fed in the agricultural land, which was the source for the heavy metals detected. Plumage color was affected in the birds residing in the urban landscape. This could be due to the lead concentration, as it was the highest metal out of the metals detected in the landscape. And the main reason for lead is vehicular emission, which is the highest in the urban landscapes. It is evident from literature that blood parameters are affected with the increase in heavy metals. And the highest aggression percentage was reported in urban landscape irrespective of low heavy metal accumulation. From this, it is understood that aggressive behavior cannot be correlated to heavy metal accumulation, and further studies need to be carried out to find the reasons. So, the take-home message from this study is that heavy metals are not attributes of urban landscapes. They can also be found in natural landscapes. Therefore, it is important to minimize the use of heavy metals containing substances and make friendlier choices. I take this opportunity to thank the financial assistance provided by the Open University of Sri Lanka from the undergraduate research grant of 2021 and all those who have listened to me today. Thank you.
Yeah, thank you, Ms. Saleh and the team for your presentation. Now the presentation is open for discussion. Any questions from the participants? It's a very interesting study. No questions from the audience, actually. Yeah. It's interesting to note the results, like the forest patch, the birds are having the very high amount of heavy metals. Like, so you gave the reason saying as the nearby paddy fields or the using various pesticides, fertilizers, and those things like.
Yes, madam.
So have the other researchers in the literature they reported the same, like, because especially the heavy metal like lead being an air pollutant, like, so did you get all three heavy metals? I think you got almost the same, like in the different environments like urban, semi-urban, and the natural. Everything is high in natural. Could you explain me any kind of like reason other than you said for lead or something like?
Yes,
I
madam.
have that just for
So,
my clarification. Yeah.
okay, madam. So, the area that covers the IOURS is
Yeah.
comparatively small than the urban and the
Ah,
semi-urban, because we have taken a larger area. So probably
yeah.
when we compare the area, the size of the selected location with the forest,
Yeah.
might be that should be why we got a higher, I mean, higher
Maybe,
results for
yeah,
heavy
true,
metals.
yeah. Very a good answer, really. That could be the main reason, like though we thought it's a forest, like, yeah. It's otherwise it's a very nice presentation and very good study. Yeah, let's thank you very much, the Sally and the team, for your presentation and the research piece of research work. Like, let's move to the other presentation, right? That is ID number 201. The green synthesis of iron nanoparticles from an aqueous extract of neem leaves, Acidicta indica. The authors are KGR Samaravikrama, UGS Vijaypala, CAN Fernando, right? So the presenter would be KGR Samaravikrama. Over to you, Mr. Samaravikrama or Mrs.
Hello everyone, my name is Umesh Samaravikrama. I am from the University of Mortua. My research topic is green synthesis of iron nanoparticle from aqueous extract of neem leaves. First of all, I explain in my content of my presentation, introduction, research objectives, methodology, research and discussion, conclusion, acknowledgement and references. Then I describe in introduction of my research. Green synthesis is a function that replaces the reducing agent with the natural extraction from plant materials such as leaves, flower and seeds. The green synthesis of iron nanoparticles does not require high pressure, high energy and temperature condition and toxic chemical. This method is more suitable for method synthesis of metal nanoparticle using the extract of plant component. The iron nanoparticle have been used as the metal and which has non-toxic, easy produced, eco-friendly and less expensive metal nanoparticle type. Iron nanoparticle applications such as medical, wastewater treatment, coatings, nanofibers and textile applications. In this study, iron nanoparticle were synthesized for use in the aqueous extract of neem leaves as a reducing agent. The neem plant is native to Indian subcontinent and dry areas throughout South Asia. It has been found in countries of Asia, Africa, and numerous countries in South and Central America. The neem plant has long been used in Ayurvedic, folk medicine, and cosmetic applications. Then I explain in research objective of my presentation. First one is that to green synthesize iron nanoparticle using the aqueous extract of neem leaves. The second one is to do characterization process of synthesized iron nanoparticle using the scanning electron microscope analysis, energy dispersive X-ray analysis, XRD diffraction analysis, UV visual spectroscopy analysis. Next, I explain the methodology of my research. First one is the preparation process of aqueous extract of neem leaves. Firstly, we get the fresh leaves, and the fresh leaves need to wash and clean well, and that fresh leaf need to dry. Therefore, use the shade dry process in few days. After dried neem leaves need to grind it process and make the crushed leaves. The crushed neem leaves from the 10 grams were mixed with the 100 milliliter of distilled water, 1 to 10 mLR, and mixture was heated at the 6 to 70 Celsius for 60 minutes. The mixture, after 60 minutes, we can get the extraction mixture, and extraction mixture need to filtration process and separate the extract, and the extract need to cool down at the room temperature. Then I explain in synthesis process of iron nanoparticle that for use the extraction from neem leaves and 0.1 m ferric chloride solution. The 0.1 m ferric chloride solution was added dropwise to aqueous extract of neem leaves, MLR 2 to 1, with stirred the mixture at 300 rpm. After the mixing process, the stirring process done by continuously for 60 minutes. After 16 minutes, we can get the iron nanoparticle solution mixture. The mixture centrifuged at 5000 rpm for 20 minutes. The centrifuge process done by 2 to 3 times. After the 2 to 3 times, discard supernatant and collect the precipitator. The precipitated solutions need to clean well. Therefore, use dialysis process. The dialysis process after the need to dry well in vacuum oven at 60 minutes. 60 Celsius. After 60 Celsius in a few hours, we can get the iron nanoparticle in powder form. That powder put in the seal type canister and kept in the 8 Celsius for further use. Then I explained characterization of synthesized iron nanoparticle. That for use the SEM analyzer, XRD analyzers, and UV visible analyzer. That for use the Hitachi SU6600 scanning electron microscope for SEM analysis. The XRD analysis for use the Bruker D8 focus XRD diffraction spectrometer, and UV visible characterization process for use the thermal scientific genius 10S series type UV visible spectrophotometer. Then I explained in results and discussion session that session. Firstly, I discussed in SEM analysis of synthesized iron nanoparticle. We can see the two pictures of SEM analysis. One for 1 micrometer, and second one is 200 nanometer. The synthesized iron nanoparticles are aggravated due to their adhesive nature and the monopoly of spherical shape appearance with particle size range between 20 to 40 nanometers. Then I explained in EDX analysis of synthesized iron nanoparticle. We can see the diagram of EDX analysis. EDX analysis from the determined atomic percentage of the iron nanoparticle. We can get the atomic percentages of iron, carbon, oxygen, phosphorus, sulfur, and potassium, and calcium in percentage level of synthesized iron nanoparticles. Then I explained in XRD analysis of synthesized iron nanoparticle. We can see the diagram of XRD analysis, and we can see the 4 peaks of the XRD analysis. The XRD spectrum shows the significant peaks at 200 to 24 to 24.16, 33.15, 39.27, and 40.83, with corresponding to phase parallel of 0.00012, 104, 006, and 113. And the values have established the characterization of crystalline metallic iron pairs. Then I explained in UV analysis of synthesized iron nanoparticle. We can see the diagram of UV analysis. UV visible spectroscopy analysis from the observed absorption peak range at 250 to 295 nanometer range due to the extension of the surface plasmon vibration in the iron nanoparticle, and maximum peak was observed at 270 nanometers. Then I explained conclusion of my research. The SEM images from identify the particles appearance spherical in shape with the particle size range between 20 to 40 nanometers. The XRD spectrum showed the 4 significant peaks corresponding to crystal difference of synthesized iron nanoparticle. The UV analysis observed absorbency peaks at 250 to 295 nanometers range, and the maximum peak showed at 270 nanometers of the synthesized iron nanoparticle. In this study, successful synthesized iron nanoparticle using the aqueous extraction mean leads as a reducing and stabilizing agent. My acknowledgement deed to the authors are thankful to the Open University of Sri Lanka gave this opportunity to publish the paper at conference IOURS 2022, and thankful to the University of Motua to provide the finance support under research grant number SRC 2113 to make this conference paper published. This my references, and thank you.
Yeah, thank you very much, Mr. Samaravikram and the team, for your very timely needed novel technique, the presentation on a novel technique actually. This is very useful, right? So let's open the floor for the discussion. Any questions from the participants, please? Yeah, yes, Mr. Samaravikraman, I have a small question from you. Like now, is it very much related to the plant, this technique, or can you use any other plant for this purpose? Right?
Yes, madam. In my research area, I use in 6 plants. In different that plant from the different between the particle size and particle size, or that's the reason. Particle size are different between plant to plant in iron
Different
and
plants.
yes.
All right. Yeah, when you change the plants, you get different size particles.
Yes, madam.
Right. So if
Yes.
you want a minute particles, that there's a range, no. So then you have to go for this name plant.
Yes, madam.
Okay, right. So do you think that this kind of output would be useful for the industry, like to stop the other chemical conversion?
Yes, madam. I using this is this is a small part of my research.
Right.
Then I using the textile material in dyeing process, in natural dyeing process.
Right. Very good piece of research. Yeah. Continue. Thank you so much,
Thank you, madam.
Samarekram and the team for joining with us. Right.
Thank
So
you,
let's
madam.
move our last presentation for the day for the biological sciences session, ID 141. The methods of single standard DNA synthesis and its application, a review by K. Vivekanandan, EDC Lilani, and I. Abegunavardhana, the authors. The presentation will be done by EDC Lilani. Over to you, Lilani.
Good day to you all. I'm paper ID 141, EDC Lilani from Department of Basic Sciences, Faculty of Health Sciences, the Open University of Sri Lanka. The paper title, Methods of Single Standard DNA Synthesis and its Applications. Let me move to the introduction first. In all known living organisms, DNA molecules are responsible for storing and carrying genetic information. In nature, DNA is mostly found in its double-stranded forms, where two complementary filaments interacting through base pairing and stacking are directed toward opposite directions, forming an antiparallel polymer. Although there are scalable methods for the production of double-stranded DNA, both in vitro and in vivo, equally efficient methodologies for the synthesis of single-stranded DNA would be desirable for a number of special applications. In fact, the synthesis of single-stranded DNA has become an enabling technology for modern DNA-based biomaterials. Therefore, the objective of this review is to offer background information on current and emerging technologies for single-stranded DNA synthesis along with some applications. Researchers have developed a number of single-stranded DNA synthesis methods including chemical, enzymatic, alternative enzymatic and bacterial-based method as well as modifications to improve their affinity and sensitivity. Synthetic oligonucleotides for gene synthesis are often made using phosphoramidite chemical methods either on traditional column-based synthesizers or on microarray-based synthesizers. Enzymatic methods can directly construct longer oligos from a double-stranded DNA template due to its exquisite specificity and mild conditions such as temperature and pH, but it requires additional steps to generate single-stranded DNA. Under enzymatic synthesis method, I will be talking about asymmetric PCR and terminal deoxynucleotidal transferase. Alternative enzymatic methods use isothermal amplification techniques for single-stranded DNA synthesis, which rely on enzyme activity or design primers to bypass the thermal destruction of the double-stranded DNA template. Under alternative enzymatic methods, I will be talking about rolling circle amplification, strand displacement amplification, and loop-mediated isothermal amplification. In recent decades, bacteria-based single-stranded DNA synthesis have primarily been used for specific large-scale production of pure single-stranded DNA. Column-based oligonucleotide synthesis is a four-step chain elongation cycle based on phosphoramidite synthesis chemistry including reprotection, base coupling, capping and oxidation steps. First, the dimethoxytrityl group of the nucleotide immobilized on the solid phase is detached by acid catalysis. Second, the 3' hydroxyl group of the nucleotide to be added in is activated by phosphoramidite and mixed with the tetrazole activator. The obtained nucleoside phosphite activator is 5' hydroxyl activated nucleotides undergo condensation. Third, a small number of 5' hydroxyl activated nucleotides not involved in the condensation reaction are prevented from participating in the reaction by acetylation. Fourth, oxidation of trivalent phosphodiester to pentavalent phosphodiester using an iodine solution. After addition of all nucleosides in series from 3' to 5', the obtained oligo is released. The microarray-based oligonucleotide synthesis is a low-cost oligonucleotide synthesis technology that may be used in place of column-based oligonucleotide synthesis method. The commercially available microchip-based synthesizers are based on the principle of phosphoramidite chemistry with slight modifications. The major differences between various microchip-based methods are the different mechanisms that are applied in steps of deprotection and base coupling. At present, there are light control, electrochemical, and inkjet plant printing methods. Despite the fact that chemically generated single-stranded DNA is commercially accessible and less expensive, it is restricted to lengths of 200 nucleotides, and it requires further processing to eliminate contaminants. The demand for efficient single-stranded DNA synthesis on the 10 kilobase scale is growing for a variety of applications. Therefore, enzymatic methods can be used to synthesize longer oligos from a double-stranded DNA template. The first method under enzymatic synthesis is asymmetric PCR. Here, PCR is carried out with unbalanced molar ratios of forward and reverse primers. The lower concentration of primer is absorbed into double-stranded DNA while the asymmetric PCR continues. In each cycle, the higher concentration of primer is employed to generate an excess of single-stranded DNA. Asymmetric PCR results include both single-stranded DNA and double-stranded DNA. Non-denaturing polyacrylamide gel must be used to separate asymmetric PCR products, and single-stranded DNA must be eluted from the gel. Terminal deoxynucleotidal transferase is a member of the polymerase X family. Unlike typical DNA polymerases, it is template-independent DNA polymerase that catalyzes the incorporation of deoxynucleotides at the 3' hydroxyl terminus of DNA, accompanied by the release of inorganic phosphate. Let's move to alternative enzymatic methods. The first one is rolling circle amplification. Rolling circle amplification is a method of isothermal amplification of circular DNA molecules. This assay involves DNA amplification using a DNA-5 polymerase with strand displacement activity to extend a single or multiple primers annealed to a circular DNA template. The strand displacement activity allows the newly synthesized DNA template to displace the previously generated DNA molecule, releasing single-stranded DNA. This enzymatic process of primer extension combined with DNA strand displacement generates a long single-stranded DNA containing a repeated sequence complementary to the circular DNA template. Next one is strand displacement amplification. Strand displacement amplification is an isothermal technique that relies on a restriction enzyme and an exonuclease deficient DNA polymerase. The restriction endonuclease will nick the target DNA, allowing DNA polymerase to extend the 3' end. This technique does have some limitations, including lower primer specificity. The last one is loop-mediated isothermal amplification. This method uses 4 to 6 primers recognizing 6 to 8 distinct regions of target DNA for a highly specific amplification reaction. A strand displacing DNA polymerase initiates synthesis, and 2 specially designed primers form loop structures to facilitate subsequent rounds of amplification through extension on the loops and additional annealing of primers. DNA products are very long and form from numerous repeats of the short target sequence connected with single-stranded loop regions. Bacteria-based single-stranded DNA synthesis is used for large-scale bacterial production of pure single-stranded DNA using modified plasmids 17 or helper M13 E. coli strains in combination with phage mids. Bacteriophage M13 infects E. coli and then replicates to produce progeny phages that exclude directly into the culture medium without causing bacterial lysis. The progeny phages are then extracted and the single-stranded DNA genome is purified and ready to be used as a source of single-stranded DNA. Single-stranded DNA is widely employed in the fabrication of nanostructures. DNA origami combines a long single strand of DNA with hundreds of short oligos to form a specific conformation. The resulting DNA structures can be used in applications ranging from nanorobots to control drug delivery. Scaffold DNA generated via rolling circle amplification is used to fold structures such as DNA nanowires or plates, which can be used as passive carriers for intercalating medicines. For more than 20 years, single-stranded oligonucleotides have been employed as a medical technique in drug delivery as well as in production of vaccines. Asymmetric PCR is one of the most extensively used methods for the direct production of short single-stranded DNA aptamers. This study detailed the typical techniques utilized for single-stranded DNA synthesis. Chemical synthesis provides a powerful tool for the synthesis of custom single-stranded DNA sequences without template. Enzymatic methods produce single-stranded DNA fragments ranging in size from several hundreds base pairs to more than 10 kilobases. However, some enzymatic methods like asymmetric PCR require additional purification steps. Bacteria-based synthesis produces milligram-scale yields of single-stranded DNA which could be boosted further using bioreactors for large-scale productions. Each procedure and alterations has different benefits and downsides, but as technology advances, single-stranded DNA will be synthesized more efficiently and used in variety of applications, providing more benefits to humanity. And this is the end of my presentation. Thank you.
Thank you very much, Ms. Rilani and the team for a very informative review, right? Since the presenters are not available for question and session, right? So let's wind up the today's session. Before winding up, let me summarize what we listened really up to now. We started with the antibacterial properties and then went on molecular docking and the the diverse group of research within the theme of biological sciences and changes in food production, and then the very important area on the malaria vector and with lot of data, and then the bionomics again. Then the last part of our session really dealt with the biomonitoring of Anopheles, the yellow-billed pebbler, and then the green synthesis, the novel technique, and lastly with the review about the single-stranded DNA sequence synthesis, right? So we should be very happy that we were able to listen to very novel and very informative techniques and the new methods and new findings in the line of biological sciences. So let us wind up today's session by thanking all the participants for their enormous support in providing the necessary presentations on time and giving us the support to continue with International Research Sessions 2022. Finally, thank you so much for all the participants, right? So let's wind up the today's session, right? Thank you.
After completion of my 2-year diploma in agriculture program, I entered the OUSL BIS agriculture program for the continuation of my further education. As a person who engaged in the field of education management and industry training in Sri Lanka, I serve my valuable knowledge for OUSL as a visiting academic and industrial expert on a regular basis. Highly experienced, well-behaved and well-qualified staff members at my OUSL life. I think it was a very good opportunity to the continuation of my education under the field of agriculture.
I was able to understand, experiment as well as gain the most of advantages of open distance learning model offered by the Open University. So with this model, I was able to learn myself, experiment things by myself and make that theories into practical as an engineer. So in that way, the academics in the Open University as well as the facilities like laboratories, library and all the other academic staff and non-academic staff was very helpful to make our journey a success.
Began the journey with Open University soon as I finished my A-levels. Industrial exposure we got from Open University is priceless. Knowledge I gained was able to make me who I am today. And the theoretical knowledge and the technical knowledge was very beneficial and influential. Today, I'm a successful woman in my field, in my career, thanks to the degree I obtained from Open University.
Good afternoon. You all are warmly welcome to the International Open University Research Sessions IOURS 2022. You have joined the third online parallel session of the online session number 3 of the day 1 of IOURS 2022. Today, you have got to discuss nice, interesting research findings under the theme agriculture. First, let me introduce the chairperson, Dr. Buddhika Arugoda, one of the senior lecturers attached to the Department of Agricultural and Plantation Engineering of the Faculty of Engineering Technology, the Open University of Sri Lanka.
Thank you very much. Okay.
Four presentations with these paper IDs are lined up in this session. Madam Dr. Buddhika Arugoda, over to you.
Thank you very much for the introduction. Good afternoon. I would like to welcome all of you for the online session 3 on agriculture. So today this afternoon, there are 4 papers lined up for the session. As you see in the screen, so we will have, each one of you have 15 minutes, and we can have 2 to 3 questions from each paper. And I would like to have general discussion after completing all the presentations. And so let me begin the first paper titled Effect of Climatic Factors on Field Grain Percentage of Rice Varieties for Cultivation During Yellow Season in Low Country Wet Zone of Sri Lanka. Over to you, the presenters.
Good afternoon, all of you. I'm GRS Ratnasiri, the author of this research, Department of Agriculture and Plantation Engineering, the Open University of Sri Lanka. The other authors are Assistant Director Mrs. W.M.N.D. Gunathilaka, Deputy Director Mrs. M.C. Milambithanachi, Rice Research Station Labuduwa Gol, and Senior Professor Mrs. C.S.T. Silva, Department of Agriculture and Plantation Engineering, the Open University of Sri Lanka. I have followed the effect of climatic factors on field grain percentage of rice varieties for cultivation during yellow season in the low country wet zone of Sri Lanka. Introduction. Rice is the single most important crop, occupying 34% of the total cultivated area in Sri Lanka. Sri Lanka has been categorized into 3 climatic zones. There are wet zone, intermediate zone and dry zone. Rice is grown in all these 3 climatic zones. According to the rainfall variability, there are 2 cultivation seasons, Maha season and Yellow season. The climatic factors such as temperature, rainfall, sunshine, wind and humidity mainly affect for the crop cultivation. The low country wet zone received the heavy rainfall in yellow season. The southwest monsoon rain and flowering period of rice overlapped around from 9th to 20th of July. This caused to wash away pollens and spikelets. This is the one of major cause for reduce the yield in low country wet zone than the dry zone. The high temperature above 32 Celsius degrees has decreased pollen fertility, increased spikelet sterility and empty grains. The wind speed is mainly caused to remove the pollens. The combination of high temperature with low and high relative humidity recorded high spikelet sterility. Therefore, this study was designed to identify the most adaptable rice varieties for low country wet zone of Sri Lanka due to changing climatic parameters during southwest monsoon, and identify the effects of climatic parameters on spikelet sterility and pollen viability field grain percentage, and finally the grain yield during the study period. The table showed 23 rice varieties which are used as the materials. This rice variety is used according to the 2.5 months, 3 months and 3.5 months time duration. Methodology. This research was conducted at Research Field Rice Research Station Labuduwa Galle, Sri Lanka. Crop establishment was 2021 in yellow season. There were 23 rice varieties as the treatments and 2 replicates. RCBD and 2-factor factorial design used as the experimental design. SAS 9.1 is the data analysis method. These 23 rice varieties grown in 2 planting dates. These planting dates were calculated according to the flowering period and the period of maximum rainfall received in yellow season. First flowering period on 1st July to 15th July. Second flowering period on 15th July to 13th July. The space between 2 plants and 2 rows in one variety is 15 cm. The space between 2 varieties is 30 cm. As the plant measurements, we collected 16 measurements of the plant such as plant height, culm height, number of panicles per plant, length of panicles, length of fruit, width of fruit, length of 5 seeds, width of 5 seeds, dry weight of plant, width of panicles, length of panicles, weight of empty seeds and filled seeds, weights of 1000 seeds, and field grain percentage. As the weather parameters, amount of rainfall, daily maximum and minimum temperature, relative humidity, wind speed, and evaporation were collected. Results and discussion. Number of panicles per plant. The significantly highest number of panicles per plants were recorded in BG 251, BG 300 and BG 352 among the 2.5 months, 3 months and 3.5 months rice varieties respectively. 2.5 months, 3 months, 3.5 months significant highest value showed blue, yellow and pink color respectively. Weight of 1000 seeds. BG 251, BG 310 and LD S2B7 varieties showed significantly highest weight of 1000 seeds in 2.5 months, 3 months and 3.5 months varieties respectively. Field grain percentage. BG 252, BG 314 and BG 359 varieties showed significantly highest field grain percentage in 2.5 months, 3 months and 3.5 months rice varieties. The first graph showed the rainfall distribution in flowering period from 1st July to 13th July. You can see the first flowering period received the high rainfall amount than the second flowering period. The second graph shows performance of rice varieties with field grain percentage. You can see the planting date 1 rice variety shows low performance with field grain percentage. Rainfall affected in flowering period of 23 rice varieties in planting date 1. 80-353, 80-362, 80-354, BG-314 varieties were most sensitive to the rainfall. Rainfall only has shown significant parameter in the stepwise regression and those were indicated in following equation. Planting date 1, grain filling percentage equals to 0.249 multiple rainfall plus 6.74. Planting day 2, grain filling percentage equals to 0.294 multiple rainfall plus 2.77. Conclusion: further analyze climatic parameters and fill grain percentage with multiple regressions. Rainfall only showed significant to the fill grain percentage. Therefore, 80,353, 80,362, 80,354 varieties among 3 months and 3.5 months varieties showed sensitivity to the rainfall respectively. BG 252, BG 314, BG 359 variety shows highest fill grain percentage in 2.5 months, 3 months and 3.5 months varieties respectively. References. Thank you very much.
Now the presentation is open for the discussion. I would like to make a note here. You will have 15 minutes for question and answers. And I think it's better if you can turn on your mics and as well as the video while you are during this session of question and answer. So now, are there any questions from the audience? Ms. Ratnasiri, now actually all these varieties are pre-tested varieties, already tested in these rice research stations. So what made you do this kind of study again?
Actually, madam, in low country wet zone, southwest monsoon rain is the major problem to the reduced yield. Therefore, Rice Research Station conducted this research to find the most suitable rice varieties for the yellow season.
Only in the low country wet zones, that's all. So you are not highlighting here the climatic changes.
All the climatic data and changes used for the analysis part, not mentioned in this presentation.
But it's shown that rainfall is effective for certain varieties.
Yes.
In the graph, I think you can see that one.
Only the rainfall effect for the fill grain percentage of the rice varieties.
Any other questions? If there are no questions, we will thank you very much, Mr. Rupa Singh, for your presentation and your information given us regarding the rice varieties suitable for the yellow season in low country wet zone, and the information given. Thank you very much. So let's move on to the next presentation, and next presentation will be on rice yield prediction in northern and eastern province of Sri Lanka using weather data.
Hello everyone, I am going to present in the title of Rice Yield Prediction in the Northern and Eastern Provinces of Sri Lanka by using weather data. Myself, Krushika Jayachandran from Department of Computer Science of Faculty of Natural Sciences of Open University of Sri Lanka, representing the paper ID 27. Let's move into the first slide regarding introduction. The agricultural sector is the backbone of the national economy of Sri Lanka as it consists of 21.7% of the total national exports. According to the national GDP of Sri Lanka, agriculture sector contributes for 7% and main food crop among them is rice. Climatic conditions prevailed in each year play a vital role in productivity. Sri Lanka is a country which entirely depends on rice as a main source of food, as national food of Sri Lanka is rice. Diverse factors influence the production of rice in Sri Lanka, and those factors should be investigated to predict and manage future demands. Sri Lankan farmers face several hazards due to biotic and abiotic stresses, and among them, unpredicted climate change is crucial. Every year, a large amount of rice production is affected due to floods, high temperature, wind speed, and several other weather elements. To supply the whole country with rising demand for rice, and there's a need for prediction of paddy yield in the near future years. This study proposes a model which represents the trend of yield in the next years corresponding with the regional climate. This study aims to investigate the trend of rice yield production in the northern and eastern provinces of Sri Lanka by analyzing the corresponding regional climatic data. In addition, this study is focused on identifying the weather elements which affect the paddy growth in Sri Lanka. The study was conducted in Jaffna, Vavuniya, Mannar, Batticaloa and Trincomalee districts based on the collected yield data during the Jala and Maha seasons. The raw weather data of the past 11 years was collected from the Department of Meteorology, Sri Lanka. Regional rice production details were collected from the Department of Census and Statistics. Research objectives were: an early study investigated the factors which affected paddy yield in the Anuradhapura and Ratnapura districts in 1984. A weather forecasting model is proposed for dry and wet zones of Sri Lanka was in 2002. A data mining and machine learning based model is proposed to predict the weather elements in India in the year of 2017. The impact of climatic change in the Sri Lankan agricultural sector was also investigated in 2005. These studies either predict or investigate the weather elements and rice production separately. The motivation to do this research lies here: to investigate the trend of rice yield production in Sri Lanka by analyzing regional climatic data, and the main objective of this research is identifying weather elements that influence on paddy growth and to verify whether future demand and supply of rice will meet or not. Using this one, government would be able to plan alternative means for paddy cultivation which adapt methods that doesn't depend on climatic conditions prevailing. This prediction may encourage farmers to engage in cultivation of paddy if the trend shows down. And government could be able to implement policies to reduce actions that make negative impact on the climate change. Let's move into the process of our study. As the steps, flow of work indicate data collection, data cleaning, pre-processing of data, correlation, building the model, training and testing the model, and feature scaling, regression and prediction. As the first step, data collection, yield data of Recently past 11 years of both seasons, Maha and Jala are taken from Department of Census and Statistics, which is a secondary data repository. Maha season of Jaffna and Jala and Maha seasons of Vavuniya, Mannar, Batticaloa and Trincomalee. Weather data was collected from Department of Meteorology, Sri Lanka, as Sri Lanka experiences rainfall for 2 seasons, northeast and southwest monsoons. Sowing methods of paddy varies according to it each year. Maha season depends on the northeast monsoon and Jala season depends on the southwest monsoon. Sowing months of paddy for each district, Jaffna, Mannar, Vavuniya, were collected from the Provincial Department of Agriculture of Northern Province. As it takes 106 to 116 days for a paddy crop, averagely weather data of 4 months were used for a season. After collecting, there is a necessity for the process of cleaning data arises. As paddy is a 105 to 115 day crop, as average weather data for 4 months were taken for the calculation. Sum of rainfall of 4 months, average of temperature and humidity of that particular 4 months. A whole CSV file was created with comprising all records of the 5 districts of all seasons. To examine the correlation of factors employed here and to select which factor is mostly influencing on yield among rainfall, temperature and relative humidity, and separate files of each district for each season used to predict the yield with corresponding climate for that region in next year. And when using the sample data, sum of rainfall of 4 months of the Maha season, average of temperature in degrees Celsius, net yield of that season in metric tons, showing months of each year changes with the start of raining, as average Maha season falls from October to January and Jala season falls from April to July. In the implementation step, as the pre-processing, checked for the categorical variables and checked for null values. Search of categorical variable, as all the tuples found with the numerical values, encoding is unnecessary, and try to discover null values in case of detection of null values to replace it with the mean value of the column. And to identify the correlation based on the observation of the result obtained, rainfall and relative humidity had positive correlation and temperature had negative correlation. Climatic factors are included in the independent variables and yield in dependent variable. And for the feature scaling, the weather and rice yield data samples were collected and measured were differing in ranges and scales. Rainfall measured in millimeter was in the range of 0 to 1000. The temperature was measured in Celsius and it between 25 degrees Celsius to 35 degrees Celsius. Average of relative humidity lies between 50 to 100. Rice yield were varying from 250 to 100,000 metric tons as cross harvest for the district. Therefore, there should be a standardization technique is used to scale the weather elements and corresponding rice yield values. Regression. The proposed linear regression model was trained with the collected data samples. The data set was divided into training and testing sets with a ratio of 70% and 30% respectively. The effectiveness of the proposed model is measured using root mean squared error. It is the square root of mean squared error. Prediction. The weather and rice yield data samples collected were measured in different ranges and scales. And standardization technique is used to scale the weather elements and corresponding rice yield values. In the results and discussions, 74 weather element and element data and corresponding yield data have been used to train the prediction model. The remaining 24 data sample pairs used for testing. Training and testing data were obtained from both Yala and Maha seasons with equal proportions. The proposed linear regression model predicts the corresponding rice yield values for 24 test samples. The proposed model showed 0.86 root mean squared error based on the prediction of rice yield values. Further, the proposed approach showed the 0.71 R-squared value based on the fraction of predicted values. To verify the correlation, the call CSV taken for analyzing correlation among the parameters employed. The heat map is illustrated here. Basically, the correlation of two variables lies between -1 and +1. Rainfall shows the 0.46 positive correlation depicts what whenever there is an increase in the rainfall, there will be increase in the yield. For degrees, vice versa. Similarly, relative humidity also shows 0.35 positive correlation, but temperature shows 0.25 negative correlation. It defines whenever there is increase in temperature, the yield become reduced. And according to the results obtained, for important features, rainfall plays the main role in the yield of paddy. Second is temperature and next is relative humidity. And root mean squared error and R-squared matrices are used to measure the performance of the model. Based on the experimental results, proposed model able to predict the rice yield values with 0.86 root mean squared error. And this study, as the conclusion, this study used a model to predict the rice yield using the weather elements in northern and eastern provinces of Sri Lanka. Weather and yield data were collected and pre-processed. Linear regression is used to predict the yield values, and root mean squared error and R-squared matrices are used to measure the performance of the model. Based on the experimental results, proposed model able to predict the rice yield values with 0.86 root mean squared error. In addition, the results showed that the correlation between the paddy growth and weather elements, rainfall, relative humidity and temperature are plus 0.46, plus 0.35 and minus 0.25 respectively. On this basis, rainfall positively influences the paddy yield while the temperature negatively influences. This prediction study might be beneficial for farmers as it predicts the future demand of rice based on the weather changes and identifies the correlation between paddy growth and weather elements in the local regions of Sri Lanka. As the limitations and recommendations, there were no proper records of showing month of details in the Provincial Department of Agriculture, and each province have an institute in the name of Provincial Department of Agriculture, but mostly last 10 years details only present as computerized data, and no efficient searching of data in the manual data entry. For the recommendation, I recommend to extend this research for whole Sri Lanka and yet with more weather elements. And thank you.
Now the presentation is open for the discussion. For the current situation for food security, this kind of predictions will be an important because northern Sri Lanka, northern eastern provinces are doing great job in producing rice yield. For you to ask questions from her to clarify. Mr. Chandran, why do you think there is a negative correlation between temperature effects to the yield? What is the biological or any kind of explanation regarding that?
Can you repeat the question?
Mr. Chandran, can you switch on your mic as well as turn on your camera, please?
Yeah, my camera and mic sound.
Can you hear my question?
Yeah, I can hear.
So, why do you think that you said there is a negative correlation between temperature and the yield? So, what is the scientific background behind this?
Yeah, temperature is needed for the yield, needed for the paddy growth number. But if it is increased beyond that limit, it becomes the crops will become burnt like that, more drought like that it becomes. So it can be a reason for the reduction.
As you said, we can do the same study throughout the Sri Lanka to get this kind of predictions, no? So do you have any idea to widen your research?
Yeah.
Area-wise?
Yes, for the MPhil study, I hope to extend this research because at that time I couldn't afford that much cost for the data parameters, the weather parameters. So I hope to extend this for my postgraduate studies.
Your sound is actually not clear. It's not in a flow. I think we are getting disturbing with some the network problem, I think. Your answer is not clear.
I said I'm hoping to extend this research for my postgraduate studies because in my undergraduate time I couldn't afford that data expenses for that weather parameters.
There also you want to do the research through the secondary data you obtained from the Department of Agriculture?
No, I have somewhat compared the data in the secondary data and from the primary sources also. I have got the data from agriculture, PDA office itself, and after that I compared it with the census website also.
So you said actually in your conclusions, the date of starting the planting is important for the study.
Yeah.
So you have not particular data about that as it seems.
No, that also I got from Provincial Department of Agriculture. Myself, I went and asked from them which months they started showing sheets of paddy. Every year it somewhat changes. Some years it will be accurately start in April. Sometimes it starts in the May, like that it changes. So for that year, I have calculated the weather parameters for that April. Other years from May, like that I have changed.
Any other questions from the online audience? If there are no questions, thank you very much, Mr. Chandran, for your nice presentation. So we will move on to the third presentation today listed, a comparative analysis of experts' opinion on the impact of climate change on tea cultivation in Sri Lanka.
Hello everyone, I am Purnima Tennakol under abstract number 29. What is the first thing that comes to your mind when you are thirsty? Other than water? Definitely, it would be a hot cup of tea. Over 155 years, the name Ceylon tea had become synonymous with the world's finest tea, and tea is one of the most important cash crop in Sri Lanka. With that, assessing the expert opinion of climate impact on tea cultivation in Sri Lanka is a timely valuable study along with the change in climate. This is the outline of the study. Moving on to the introduction, let's see some important statistics like the tea cultivation extent and the people who employed in Sri Lankan tea sector. When it comes to the economics of tea, it is a key contributor to Sri Lankan economy in terms of foreign exchange earnings, employment and food supply. Sri Lankan tea industry performed lower yields and higher cost of production by showing a downward trend in the Sri Lankan tea production, even though the world tea production has an upward trend. Left side graph clearly shows the fluctuations in Sri Lankan tea production from 2011 to 2020. The peak production of 340 million kilos has been observed in 2013. Since then, there has been a gradual decline. This caused a lower productivity too. Let's move on to the problem. Declining the tea production and productivity can be identified as the main issue. There are many reasons for this. Among them, climate change impact is a key reason. Expert perception on various climatic factors have been considered and checked whether there is any significant difference between the opinions among three expert groups. The objectives of study are to compare the perception among different tea expert communities and evaluate if whether there is any significant difference among expert perceptions on climate impact which would affect the production, productivity and profitability of the tea sector in Sri Lanka, and also to suggest the most feasible climate change adaptation strategies and management protocols on the Sri Lankan tea culture. Moving on to the methodology, primary data were collected from January to March 2022 from 41 respondents, including 10 executives, 25 estate managers, and 6 academics. Data were gathered by using key performance interviews and focus group discussions by using a semi-structured questionnaire. The gathered primary data was analyzed by using analytical hierarchical process, which is also called AHP, and it was statistically analyzed by multivariate regression analysis. A pairwise comparison was done by using 9-point scale and obtained the following criteria weights. Consistency ratio was calculated by using the criteria weights, and ranking was done according to the degree of importance. Moving on to the most important part of the study, that is the results and discussion. According to the results of AHP, or the analytical hierarchical process, all three expert groups have ranked rainfall as the first priority. Then the practitioners, that means the executives and estate managers, have ranked temperature for the second place, while academics have ranked soil pH as the second priority. As the third priority factor, three expert groups have ranked for three separate climatic factors by showing a difference in their perception. According to the coefficients which obtained by multivariate regression analysis, rainfall and temperature highly prioritized, since those two factors have the highest coefficient value among 10 climatic factors. And estate managers have highly ranked than other two categories. When considering the soil depth, soil pH, gravel content, and sunshine duration and solar radiation, academics have highly prioritized than practitioners. Rockiness and soil type are highly ranked by executives than other two groups, while wind and relative humidity is highly prioritized by estate managers. It can be concluded that higher degree of importance given on the rainfall and temperature by all three expert groups: soil pH, soil depth, gravel content, and sunshine duration and solar radiation were ranked by academics, while rainfall, temperature, relative humidity, and wind were prioritized by estate managers. According to the expert perception, developing tolerant cultivars, practicing good agronomic practices, conserving soil moisture, promoting climate smart tea production, adapting rainwater harvesting techniques, selecting most appropriate areas for tea cultivation, and minimizing human activities which affect the climate badly are the adaptation strategies to minimize or mitigate the climate impact on tea cultivation. The importance of this study is very much significant to improve the sustainability of tea sector in Sri Lanka. Since the experts are the top level decision makers, this study provides useful insights to policymakers in developing corporate strategies for the mitigation of climate change impacts faced by tea sector at present context as well as in future context. Let's move on to the recommendations. So, there should be a common agreement on most significant climatic factors with relevant to the priorities among three expert groups, and it is necessary to decide suitable adaptation and mitigation strategies based on the prioritization. In order to sustain the tea sector, the suggested adaptation strategies should be practical and financially viable, reducing the cost of production. Strengthening the research and development activities and provide good extension services could be recommended as the recommendations. So these are the references which I use for the study, and this is the end of my presentation. Thank you for your
Now the presentation is open for the discussion. Common problem today, the climatic change and their effects on all our crops, not only the tea actually, but as the tea, which brings us the dollars to Sri Lanka, earnings to Sri Lanka. This is very important to study and take decisions on what is the remedies actually we can give regarding the tea cultivation if the yield drops. Any questions from the audience? Mr. Nakon, you can open your mics, and if you like, you can turn on your video also. In your recommendation, you have mentioned extension services has to be regularized. Right? So how do you think the extension services are linking towards this climate changing effect?
Actually, ma'am, that I think we can instruct the tea growers as the extension officers related to this tea sector. We can advise or instruct these tea growers how to practice these common adaptation strategies, which can, I mean, the adaptation strategies to minimize the climatic changes, like following good, how to follow good agronomic practices and soil moisture conservation practices, and promotion of climate smart tea clones. So that kind of knowledge can be given by the extension through a extension service, because for the tea growers. So I think that would be affect the contribution of this extension officers. So the extension services maybe affect this climate change impact.
This actually strengthening the extension services with correct knowledge is a somewhat reachable. That is, there it has a reachable point to solve this problem up to some level. So,
Yes.
is there any other questions from the audience? If there are no questions, we'll wind up the presentation. Thank you very much for Mr. Nakon for your nice presentation.
Thank you, ma'am.
Let's move to the final presentation today in this session. Studies on growth performances of cinnamon in cocoa peat as nursery pots.
Good evening. The research project I am presenting today is Study on Suitability of Cocoa Peat Pellets as Nursery Pots for Cinnamon. I am PKD Pabasara. This is the content of my presentation. Introduction. Cinnamon. The outer bark of cinnamon plant is a very popular and unique spice in the world. For true cinnamon or Ceylon cinnamon, Sri Lanka holds the monopoly power in world market. With the increasing demand and growing market for cinnamon, there is an increasing number of new growers entering into the cinnamon cultivation. This shows how the land area under cinnamon is expanding in Sri Lanka. From 2010 to 2020, there is a 15.5% of expansion in land area under cinnamon. To keep up with this expansion, there should be a continuous nursery plant supply. Thereby, there is a requirement of a stable nursery plant production. In a nursery, potting media has an important role. Recommended potting media for cinnamon includes topsoil, sand, coir dust, and cow dung. But currently, there are difficulties in procuring these materials due to legal boundaries, high cost, low availability, and environmental limitations like factors. In this situation, cocoa peat can be a possible alternative to current cinnamon nursery media. Cocoa peat is a non-fibrous, spongy, lightweight coffee material that holds the coir fiber in coconut husk together. After going through a manufacturing process, different types of cocoa peat products are produced. Cocoa peat pellets is one of them. According to previous literature, various forms of coir has been tested and accepted as growing media for several crops. Therefore, this research study was initiated to investigate cocoa peat pellet as a possible growing media for cinnamon. Objectives. General objective to identify the cocoa peat pellet nursery media and fertilizer schedule to enhance the growth performance of cinnamon seedlings. Specific objectives are to study the growth performance of cinnamon seedlings in cocoa peat pellet as nursery pots, and to determine the fertilizer requirement of cinnamon plants at nursery stage in cocoa peat pellets as nursery pots. Methodology. This study was conducted in National Cinnamon Research and Training Center from 2018 to 2021. Seeds of Cinnamomum selenicum were used as planting material. Pots were arranged on a wire mesh table. Uniform shade and uniform irrigation was maintained. Albate solution was used as the fertilizer. Fungicides and pesticides were applied when required. This project was conducted as two experiments. Experiment 1, 2-factor complete randomized design was used. There were two factors. Factor 1 is the nursery media type. Here, There were 3 levels, A1, 200 into 120 mm size polybag filled with department recommended nursery mixture. A2 is 50 into 60 mm size cocoa peat pellet. A3 is 60 into 120 mm size cocoa peat pellet. Second factor is the concentration of liquid fertilizer or Albert solution. 3 levels are B1 0 gram per liter, B2 37.5 grams per liter and B3 75 grams per liter. Fertilizer was applied as a foliar application starting from 2 months after planting and continued at a 2 weeks interval. With 3 levels for 2 main factors, there were altogether 9 treatment combinations and 4 replicates per each treatment. Growth data were recorded at a 3 weeks interval. Experiment 2. There were 9 treatments arranged in a complete randomized design with 4 replicates per each. From 9 treatments, first one, A1, is the recommended mixture and for other 8 treatments were different concentrations of Albert solutions applied to the plants grown in 60 into 120 mm size cocoa peat pellets. So these concentrations were 0, 0.5, 1, 2, 3, 4, 5 and 6 grams per liter concentrations. Fertilizers were applied directly into the pellet starting from 2 months after planting and continued at a 2 weeks interval. Growth data were recorded in a 1 month interval. Data collection and analysis. Number of leaves per plant, shoot length, root volume, shoot dry weight and root dry weight were recorded as growth data. In experiment 2, leaf area and leaf color were recorded. Data were analyzed using ANOVA procedure. Results and discussion. In experiment 1, media types and fertilizer levels were compared to select the best performing treatments. Here, mean values of growth data at the end of the nursery period, 6 months after planting, are presented and mean separated at 0.05 significance level. There is no significant interaction effect between 2 main factors of media and fertilizer level. When considered the main factor media type, there are some differences. All the growth data are lowest in A2, the smallest pellet size. Recommended media has the highest number of leaves and shoot length, while A3 is similar to recommended in shoot dry weight and has the highest root volume and root dry weight. In the second factor, fertilizer level. Here only the number of leaves shows a significant difference. Other parameters have significant similar values at 3 levels. All the growth parameters showed not much response to the fertilizer level. Plants without fertilizer didn't have the healthy greenish leaf color compared to the recommended media. Plants in 50 into 60 mm size cocoa peat pellets showed significantly poor performance than plants in 2 other media. Also, plants' root system was coiled and tangled around the netting. In overall, it can be said that plants grown in 60 into 120 mm size pellets had similar performances to the plants in the recommended potting media. Since the foliar application of fertilizer didn't show a good result, in experiment 2 fertilizer was applied into the pellet. So in here, in number of leaves, root volume, shoot dry weight, root dry weight and leaf area at the end of the nursery period, there is no significant difference between the levels.
but fertilizer levels of 4, 5 and 6 grams per liter have performed better. This is how the shoot length differs. Other than the recommended media, F4 and F5 showed higher shoot lengths. Although not significant, from plants grown in pellets, highest number of leaves, shoot length, root volume and shoot dry weight were recorded in 4 grams per liter fertilizer level. Leaf color variation with applied fertilizer concentration. Lowest fertilizer levels had yellowish leaf colors, but 4, 5 and 6 grams per liter fertilizer levels had resulted leaf color of moderate olive green, which is similar to the leaf of plants grown in recommended potting media. Considering all these factors, 4 grams per liter level can be selected as the suitable fertilizer level with plant growth performances similar to the recommended media. Conclusion: 60 into 120 cocoa peat pellets can be used as nursery substrate for cinnamon seedlings, with application of 4 grams per liter Albert solution in a 2 weeks interval and applied directly into the pellet. These are the references I used. Acknowledgement. Thank you for your attention.
Now the presentation is open for the discussion. I think you want to solve a problem in the nursery of cinnamon, making cinnamon plants. So thereby using pellets. Pellets. So why do you use two sizes of pellets here?
There are 2 available sizes in the market, so I used all the 2 sizes and try to select the most suitable size for the cinnamon.
Now there can be some cost involved in this process than the normal traditional way of doing cinnamon cultivation. No? So what about the cost involved here? Because always now this is targeting the export market, high profitable levels. So they are actually thinking about the profit also very important here in this point. So therefore, do you have any idea about the cost involved? Feeds for the feeds you have to make, you have to pay, as well as for the Albert solution you have to pay. Now the price has increased a lot, all this material. So considering that, what do you have any conclusion? Well, you have anything you have gone through any information regarding this? Okay, go ahead.
Yeah, for our recommended media, it will cost around only 20 rupees.
20 rupees per plant?
20 rupees per pot.
Per pot?
For the production of a pot with recommended media. But with the cocoa peat, it will cost around 35 rupees. So cost is very high. If we, when there are some problems in acquiring the materials like cow dung, like that materials, if we have no other option, we have to go for cocoa peat, but the cost is very high. So farmers are reluctant to go for this cocoa peat pellet actually. If they have no other choices only, they can go for cocoa peat pellet due to this high cost. But the handling and labor requirement is very much low in this cocoa peat pellet.
I think so much. And also to under current situation, cocoa peat pellets, are they available freely in the market to buy?
Yeah, they are available. There are few products. Mostly they are producing for export market.
Yes.
But they are available. But cost has earlier it was 12 rupees per pellet. Now it has cost around 16 rupees.
Yes, definitely it should be according to the current situation in the country. Do you have any questions regarding her invention? Actually, today we need to find out different as well as more methodical solutions for our agricultural sector. So I think therefore these kind of innovations are very important. But you have to consider about the cost first. Any other questions from the online audience? If there are no more questions, we'll wind up the presentation. Thank you very much, Ms. Prabhasara, for your innovative presentation. So altogether, we had 4 studies today, presentations today. We have 2 papers on rice cultivation. One of them is related to the climate change and the yield of rice, while the other paper is about the prediction of yield based on weather data. All of these are actually very much informative for the today's condition about food security and the current situation we are facing in Sri Lanka, considering all the economical and other socio-economic factors. And the third paper is on tea cultivation using a questionnaire survey. That is also actually we had a lot of information about how the 3 sectors are, the predictions about the influences on reduction in tea yield, why it happens, their idea. And then finally, the 4th paper is on cinnamon testing on cocoa peat as a medium for nursery plants. Another innovative research to the current agricultural sector, where the new innovations need for improving the agriculture in Sri Lanka under the present situation. So, I think now it's time for us to wind up the session 3 of agriculture, first session of the academic sessions. So let me thank all of you who did the presentation again. Thank you very much for your informative presentation, as well as who involved in the technical support as a team here in our IOURS 2022 for your great job in conducting the online session in IOURS 2022. Thank you very much for all of you for today. Thank you.